journey despite snow and ice and what the elements have to offer for us to come in person as we asked. And I also would like to very much welcome everybody on the phones and lines from far away. We didn't make the lines available to those who we thought would be able to come because we really would like to have a dialogue, which is face-to-face communication. And for that, we ask, if it's not too late, for people to move up during the course to just be able to talk more freely and be heard, if that's possible. We really like to see your faces down here. Other than that, we're starting with RISK21. You know, this esteemed institution deals with risk every day, even if we don't do it ourselves. The work we do is differently, tangentially affected by it one way or the other. So it is important that we know what we're doing and we know and are open to other challenges and to new ways of thinking about things we're doing on an everyday basis. In that regard, we would like to use this day for dialogue, for being open-minded, for listening and hearing new things. And with that, we get started with the institutional memory that is present within us. Dr. Ron Lorenzen. Just one second, I'm going to run through that. He will give us a short, very short introduction of the lay of the land, what we have done, what we're doing, and how he sees what we might be doing and why we are challenged. Followed by that, we have a esteemed list of speakers who are introducing us to RISK21, and I will let it up to them to explain to us what that means. For some reason, my microphone comes in and out, and I apologize for that. Maybe I should switch to this one. Is this better? So our first speaker is, after Dr. Ron Lorenzen, is Dr. Alan Bubis from Imperial College London. He is one of the co-chairs to the RISK21 project, followed by Dr. Michael DeLarco from the National Institute of Child Health and Human Development. He is working on the National Children's Study and is also a co-chair for the exposure sub-team of the RISK21 project. After a short break, short break, we will hear from Dr. Angelo Moretto, who's with the University of Milan. He is a co-lead to the cumulative risk subcommittee of the RISK21 project, and from Dr. Tim Pasteur, who's with Syngenta Crop Protection. He's also a co-chair to the RISK21 project. And from 3:30 to 4:30 for 1 hour, we really want all of you to become part of this endeavor by asking and responding to questions that we are bouncing off an esteemed panel assembly, for which we have gathered Dr. Doug Wolf. He's also a RISK21 team member. We are missing one of the team members, Dr. Craig Rowlands, who regretfully is lost in the snow due to his flight cancellations. We are very sorry about that. But we have Dr. David Jacobson-Cram from FDA CEDAR, who will lend us a drug perspective on the materials we hear today. Dr. Dennis Keefe from OFS, Dr. Clark Carrington with CHAT, and Dr. Doug Karras, who's with Risk Communication. So I hope you will enjoy it. Use the opportunity to open your minds to new ways of thinking and doing things. And what we would like to develop is an enthusiastic step forward into a workshop that we hope to hold later in the year, where we can apply some of the things we're discussing today. So Dr. Lorenzen, if you would want to come up and give us a brief historical perspective. The pointer is there.
How do I do the slides? He does them up.
How do we get the next Yeah.
I mean, how do I do the slides? Oh, here
Just
we
to
go.
They
transition.
are all loaded. Yeah,
Well,
there it is.
can I use this?
Yeah, you can use the space bars or the arrow keys here.
Okay.
The arrow keys.
And this is, and where's the pointer?
The pointer is the red dot here at the top.
Okay. Got it. Well, if there's any doubt, I'm on sort of a short leash here. Um, and I didn't know this was, that you wanted this to be provocative, and so I spent the weekend toning everything down. I'm reminded of the president of a religious congregation that went to a famous theologian and asked him if he would give a sermon. And he said, "We want a long sermon. How much time do you need to prepare that?" And he says, "Oh, that's no problem. I can do it the next day." He says, "Well, what if we wanted a short sermon?" And he said, "Well, in that case, I'd need a week to prepare." And that's what's really tough about this area. Slides came out all right. You know, to try to give a talk about what's been done in risk assessment or safety evaluation or all the things we've been doing for a century here at FDA is real tough. Okay, but I'm going to try to leave you with some maybe points that you may not consider and which, coming from a regulatory agency, I know are very important. The important thing, of course, is what do these words mean? What do the concepts mean? And do they age well? The answer is not really, okay? And that's why the nomenclature is a problem. This is one of my favorite quotes from Upton Sinclair. "It is difficult to get a man to understand something when his salary depends upon his not understanding it." And if I were to replace the salary, which I underlined there, with gratification, I would have everybody in the audience here in this group. What it means that is personal bias is difficult to surmount, and personal bias is really very problematical in the safety area. Well, let's go to the beginning. Over 100 years ago, Harvey Wiley put together his poison squad. Now, that's Harvey up in the, see if I can work this. There, whoops. Need some dexterity for this. There he is, the old guy. And with a bunch of kids around him. These were his employees, okay, and they made up what was later called the Poison Squad, and they're all dressed up, looking like they're going to go out to dinner or something, which they were. White tablecloth and napkins and all the equipment. They were going to dinner to eat food additives, food preservatives. Well, you know, it's not hard to understand that this was really theater and a little bit of risk assessment perhaps, but it's really theater. And here's a sort of a capture of what he did by somebody watching, and I'll read it to you because I need to say, he stopped the experiments only when the chemicals made several of the diners so sick that they couldn't function, nausea, vomiting, etc., inability to work. By this time, though, stories of the men's indigestion had run rampant and were being followed by fascinated readers all over the U.S. The table trials, as they called them, even made the minstrel shows. I thought, what's that old song about they're selling tickets to the hanging? Well, okay, this is similar. But in the end, Wiley gained a congressional hearing. Okay, and more importantly, I'll add, this helped pass the 1906 Pure Food and Drug Act, which really everybody sort of marks as the beginning of the Food and Drug Administration. Also, this publicity helped Wiley support his bias, which I talked about, his moral bias, that chemical preservatives had no place in food. Now, we're in the Wiley building, and we love our founding fathers. Okay, however, this was over 100 years ago, and things had a little different flavor then. But nevertheless, this beginning, you know, started the ball rolling, and we still have some of the trappings of it. And Wiley, of course, needed to be a crusader before being a scientist, and it's not uncommon for these new areas. At any rate, the bottom is these boys were going to get sick. Okay, Wiley designed these trials, if you will, to confirm his bias. You know, if they wouldn't have gotten sick, we wouldn't be hearing about them. I mean, so it was theater, okay, and it's not a science. It's not science. It's not objective risk assessment, but it's what we have from the beginning. What I want to say from that is that the performance of safety and risk assessment, whatever words you use to capture it, is always predicated upon a moral premise. Of course, 100 years later, as we are now, science drives the risk assessment methodology. But what we forget is that Congress defines the moral premise, and that's the safety of products that go into food. Okay. And we must remember also that moral premises are not a special purview of science or scientists. It comes from elsewhere. Not that we don't have some role in it, but largely it's elsewhere. But Wiley assumed that these premises were in his bailiwick. Actually, he was wrong. Okay. I mean, 100 years later, there are preservatives in food. Okay, and I'm not going to comment on whether that's wrong or right. It's not my prerogative to. Okay, and it wasn't his either. It wasn't his prerogative to make that assumption in the first place. This is an issue that people forget when they get deep into the science. Okay. You know, these kinds of risk assessments, they're not performed in a vacuum. They're not done for our fun, and they're not there to get our salaries, even though probably everybody in this room is getting their salary based upon them. The purpose is safety. Now, I don't know what the word safety means, but we all know what it means. That's the purpose of this. And forgetting this is a lapse that propels risk assessment technique into theater. That is, the risk assessments become biased by the convictions of the risk assessor. You know, this is hard to take if you're a scientist, that you could be biased. Oh, yes, we can. Having said that message I want to display that we should be careful of our biases and overstepping our authorities or our expertise. But having said that, if you look over the sort of the landscape of 100 years, the way I see it, there are two generic forms of risk assessment. The first one is what I'll call qualitative risk assessment. You don't see these words too much anywhere, but this is the best fit that I can make it. And this risk assessment or safety assessment or whatever you want to label it, was employed for about 50 years, starting from the beginning until 1958, at least in this agency. But it's still used today for a lot of things, okay? What it is a black and white type risk assessment, an either or, all or nothing. Something is either something or something else. It's usually two states of nature. And in this case, we could say it's toxic or it's not toxic. Or maybe something you're more familiar with, it's carcinogenic or not carcinogenic, okay? And that's it. It's one-dimensional. Okay, and the 1938 FD&C Act, which was sort of the follow-up to the 1906, all the terms are there that indicate that this is a qualitative thing. Something's poisonous, deleterious, adulterated, filth, putrid, okay, or not. Okay. There's another one there that sneaks through. It's either there or it's not. In other words, there's this whole two-state view of risk. Okay, it's risk. It's there. It's not there. In other words, this paradigm is about classification. If I had more time, and that's another session, there are agencies out there that operate almost completely upon this, is or is not. Well, reality is not one-dimensional, and neither is risk or safety. And it's a cartoon. Not as I would have liked a better one, but I couldn't find it. One that I could afford, at any rate. I don't know if you've gone to the internet and tried to get cartoons. They're expensive. At any rate, what this shows is that it's complex, and this is something we're not even, well, we are considering this today. What do we do about more than one? We only consider one thing at a time. What do we do about more than one thing at a time? At any rate. And then there's quantitative risk assessment, which I think everybody feels they're here for. This was invented by FDA scientists in the 40s and 50s. Some people make claims that they did it too. It doesn't matter. I can see what they did here. This is not discovered, this was invented, okay? And it had to be implemented for the, that's a FAA, the Food Additive Amendments of 1958 that came down from Congress. That was the deadline we had to meet.
5 minutes?
But this...
5 minutes?
5 minutes, I'm close. This integration is far ahead of its time. I think I call it a quantum leap. It's very easy to be blasé about it, but what they did was incredible, okay? And they changed the whole process for safety and risk assessment into a multi-dimensional process, in particular one that has a dose-response metric, okay? Finally, we honor Paracelsus, who 500 years earlier said the dose makes the poison. Okay. They also integrated into the whole process the past couple decades before then of laboratory animal sophistication. We're using laboratory animals for surrogates. And they pulled this all together, okay? And when it went out, you know, it made it clear that safety and risk is about dose. It's not about occurrence, okay? Occurrence is qualitative. And it got quick international buy-in. It went, as they say in the internet these days, it went viral. Real quick. You know, the quantitative risk assessment for carcinogens, which I've done much of my career, actually came a little bit later, like 1970 or so. And we always think of it as being different, but it's really not different in concept. It's really the same thing. Comes from the same cloth, if you will. but it did give us the more familiar risk language, if you will, risk idiom. So where are we now? That would normally have been just a nice launching pad to go into things, but so where are we now, and what are you going to do here? Or what are you about here today? Actually, quantitative risk assessment has not changed since the 50s in any significant way. The public and the press still emotes over detection, not the risk. You know, hardly anybody says, how much? They say, oh, it's there. And so their perception is confused and it's wary. And what has happened sort of right in front of our eyes without us really sort of seeing it is that analytical chemistry has gone viral. We've gone from parts per million to parts per quadrillion in my career. That is more than a million-fold increase in detectability. This is phenomenal. It's hard to imagine anything being so incredible. And so we are in a situation pretty much now where if we want to find something in anything, we can. And that, of course, leads to a proliferation of issues. And, you know, only recently with the REACH program and everything, we realize there's a lot of substances, chemical substances in the environment that don't have any toxicity, don't have any risk data on them. And now we have all of these things that we're finding in parts per trillion, et cetera, levels. What we have as a situation now is too many toxins, too little time, and too little money. And so we're all in, I guess, what is today what we would call faster, cheaper, better. Faster and cheaper are easy, but better or good enough is a problem. So I believe that after 50 years, again, that it's time for another quantum leap, okay, of understanding, but we need to keep the purpose of safety in mind when we're thinking about this, because if we don't, it won't happen. And it's not just taking care of these issues that I pointed out here. So, good luck.
Thank you so much for this setting the stage, and we go right ahead with the next talk, Dr. Ellen Boop from okay, right?
Okay, thank you. Yes, I know the pointer. And well, good afternoon, and I'd like to thank Susie and Sabine and the FDA for giving HESI the opportunity to present to you the current status of this activity that's been ongoing for a few years now, the so-called RISK21 project. And I thought that the presentation we just heard from Ron really highlights some of the reasons that we instituted the RISK21 project. I don't need to tell anybody in this room that we're faced with a plethora of chemicals to which we're exposed. These are substances which we deliberately administer to humans, for example, human therapeutics in the form of drugs. They could be household cosmetics, some substances we add to food as ingredients, but there are incidental exposures such as from emissions and effluent. And we shouldn't forget the world of natural compounds, the natural toxins and other chemicals in our diet. And of course, we're faced with the difficulty of trying to make sense of the exposures and the relative risk to different subpopulations. And we're now living in a dramatically changing landscape which is impinging on at least how we think about how we should do risk assessment, and thinking that it should make a difference. But how is it going to make a difference? We've seen rapid advances in scientific knowledge. I mean, the number of scientific papers and the knowledge content of those papers in the last 20 to 30 years is truly staggering. When you think of just the areas of genomics and epigenetics, we live in the era now of the $1,000 genome, and people are estimating in not very long time it's going to be $10 a genome. In other words, we can sequence an entire genome for a few dollars. Interpreting the data is another question. Allied to that, there have been really profound technological advances. I'm thinking of high throughput technologies. We're all aware of the ToxCast program, up to 700 different endpoints in a high throughput system. Computational toxicology, the Moore's law, and the expanding power and capacity of computers, allowing us to address computational questions that would be inconceivable just a few years ago. Putting that together in systems biology with the information coming out of bioinformatics strategies and high content analysis, the omics and other high content approaches. And we live in the era of big data. I mean, we're talking about millions, if not trillions, of data points, which we're trying to interpret and interrogate to get meaning out of. And people say now that the mathematicians will inherit the earth, because they're the people who are going to tell us how to make sense of these data sets, which are incomprehensible without a deep mathematical conceptual framework. We've heard already of the need to assess the risk from combined exposures, and Angelo will go into detail in his presentations. What do we actually mean by combined exposures? How broad is that question? And we've got emerging issues we're all aware of, new and exciting technologies and materials, nanotechnology, for example, global trends, climate change, biotechnology, etc. The increasing complexity of risk communication. Ron mentioned this, that it's becoming very difficult to communicate. And if we're moving towards a quantitative, transparent expression of uncertainty in our risk assessments, which is quite appropriate, communicating that to the public and policymakers has a really difficult problem. And there are societal and other demands that we move from animal testing to non-animal assessment methods, either in vitro or computational. And so the National Academy, of course, in its reports on toxicology testing in the 21st century, sometimes shorthanded to TOX21, and Science and Decisions, made a strong plea that we should invest in research and intellectual input to move away from this paradigm that served us quite well, I would say, for several years, where we use experimental animals as a surrogate for humans to identify and characterize the hazard, and then extrapolate to human risk or risk levels, to using an alternative suite of methodologies and assessment approaches which don't use animals, such as computational approaches, high throughput and high content analysis, sophisticated three-dimensional cultures and medium throughput, the DARPA project, where it's seeking to put a human on a chip with 10 tissues in an integrated fashion, all derived from stem cells, so continuously renewable and differentiated. And the ultimate goal for the time being is to integrate this into a computational model, which is quantitative systems biology. And so we're faced with this landscape, and this TOX21 is the savior of the world. How are we going to actually make meaningful decisions from this information? And this is where, a few years ago, HESI sat down with a group of scientists and said, well, we don't want to reinvent the wheel. There's a huge amount of work going on in the world. How can we bring this together somehow to try to help people make sense of it and provide a more transparent guidance as to perform risk assessment on the output of these methods? So the RISK21 project was born, and after some discussions agreed that the mission should be to bring applicable, accurate, and resource-appropriate approaches to the evolving world of human health risk assessment. One of our distinguished colleagues coined the phrase, what we really want is enough precision to enable us to make a decision. That we don't need to do the highest tier biologically based dynamic model if we're simply, when there's a 10 million-fold margin of exposure between the toxicity and the maximum possible human exposure. So the way we would do this was to convene experts from academia, industry, government, and other stakeholders. And in fact, when we counted it up, RISK21 has involved over 120 scientists, largely from Europe and the USA, from both public and private sectors. We would develop risk assessment approaches that embrace advances in scientific knowledge and methods that would utilize, to the extent possible, existing information, but find smarter ways to use that information. Revise current thinking, how to approach the science and art of risk assessment. So think about the paradigm and can we think of a better way to approach the question. And we came up, after quite a lot of discussion, with the so-called RISK21 roadmap, which is shown here. And I'll just walk you through this. So one of the things that we emphasize is that we really have to start with problem formulation. What is it that we're actually trying to solve here? We've heard about how HESI risk assessment is not conducted in a vacuum. It's to meet the needs of policymakers and society, providing scientific input based on weight of evidence. But that evaluation has to be within a context. So problem formulation sets the context. It sets things like what is the actual exposure scenario we're concerned about? What are the populations that we're concerned about? How secure do we need to be in our decision making? Are we simply prioritizing compounds, or are we authorizing something? So these are some of the questions that go into problem formulation. So that was the first key thing, that we really need to focus on defining the problem. And then we felt that we really need to lead off with exposure rather than with toxicity, as is the want with risk assessment today, that we tend to focus on hazard. In fact, Ron emphasized how even despite all the years that have gone by, we're still seeing the idea of hazard-led decision-making as opposed to risk-made decision-making, risk-based decision-making. And of course, risk is the product of hazard and exposure. So if you go to hazard-based, you're ignoring exposure. And we felt we should lead on exposure, and you'll hear Mike DeLarco will explain exactly what we have in mind there. And as with both exposure and hazard, we felt that we needed a tiered approach, which gives us this idea that we'd get enough precision to allow us to reach a conclusion, and we didn't need to invest huge amounts of time and resource when it wasn't necessary, if we could make a decision on a more conservative, lower tier assumption. And so we start with the situation where we've got minimum information on exposure, and we go up to fairly high tier levels, and you'll hear more about that later this afternoon. That would then, based on the output of your exposure assessment, direct you to what you need to do on the toxicity side. And again, this is a tiered approach, starting with, for example, de minimis values like the threshold of toxicological concern, or simple quantitative structure activity relationship methods, and moving up to more refined methods. And finally, I would have put at the top of this mode of action, which is really just one window into a systems-based toxicology. I think mode of action is a systems-based approach, and we'll see more refined systems-based approaches in the years to come. And they would form the highest tiers of the assessment. And then we would use the output of these two and array them on this RISK21 matrix, which I can bring up in closer detail. And the matrix probably needs a little bit of explanation. The first point is that it's a relatively minor one, but it's important that potency, of course, is expressed on a scale which goes from a high number to a low number. In other words, the lowest potency compounds have the highest points of departure. Obvious, but that's why the numbers are scaled in this way. We array the estimate of exposure, either a point estimate or a range along the x-axis, and a point estimate or range of estimates of toxicity along the y-axis. And whilst we've got the units in mg per kg here as per kilogram of body weight, what we would like to emphasize is that the expression of exposure and toxicity can be in any units as long as the same on both axes. And I emphasize that because it may be that we will be using estimates of internal or target tissue exposure when we get to higher tier PBPK models, for example, and we may not back extrapolate to external dose. We may want to actually compare the internal dose. And so you can use any estimates, any dose metrics, I should say, as long as they're on the same scale. The other point is that we've shaded this from a nice reddish color up in the top right to a nice light green on the bottom left. For most parts of the world, you're familiar with the traffic light system, red stop, green go. And what this really means is that something down here in the green area, which is low potency and low exposure, is very likely to be of minimal concern, whereas something in the top right, high exposure, high potency, is very likely to be of significant concern. And in the middle, we have a fuzzy area shaded in yellow, which means that that's the bit the risk assessors are going to spend some time discussing among themselves about how we interpret the position on the matrix. And of course, that will be partly contextual, depending on the problem you're trying to address. That there will be situations where you can afford to be slightly closer to this end of the scale than and others where you'd like to be really down here. It depends on the problem. So we array our exposure estimate, in this case a range, along the x-axis, and our toxicity estimate, a range again along the y-axis. And where they intersect, it gives us the zone of concern, if you like. And this shows us the relative positioning, firstly to the area of low concern or high concern. And secondly, it tells us, depending, based on the dimensions, where we would best refine the estimates. Would it be better to refine the exposure estimate or the toxicity estimate? So what would move it furthest away from the midline? And we could then do a higher tier assessment. We'd go back round the cycle, enter this or this, depending on what the decision based on your position on the matrix was, and undertake a higher tier, and then replot and see where you come out. Where does this move to? And does it give you enough precision to conclude, based on your problem, that there is little or no real concern? So we can plot ranges, but we also wanted to refine this, recognizing that one would, particularly in higher tiers, want to perform some more sophisticated analysis. And so we have also introduced the possibility of using distributions. And that would then, of course, give you a shape more like this. You can feed in the appropriate parameters, which could be either empirical observations or a modeled distribution as appropriate. And we're working on the expression of uncertainty as well, which quantitative expression of uncertainty, allowing us to plot that on the scale as well. So this is all very well, and it will be the basis of publications, but we felt to be really useful, we should introduce a web-based tool. And that web-based tool would allow you to plug in your own numbers, but it would also allow you to use prior information on exposure from lookup tables and prior information on toxicity from lookup tables. And you'll hear more about lookup tables and the way we visualize using this in the exposure assessment tiers from Mike DeLarco in his presentation. But I have to tell you that hot off the press, we are here today to demonstrate for the first time ever to an audience our web-based tool. Now, you'll hear about the whys and wherefores later on this afternoon, but just to emphasize, this is an alpha version, but it does work, and it will give you at least an idea of what we have in mind for this as a publicly accessible, freely usable web-based tool in the future. Of course, we plan to make all of the results publicly available And so we have a series of publications, starting with 2 overview papers, which have already been submitted, and then a series of papers on the exposure, on the toxicity approaches, an in vitro, in vivo extrapolation framework, and a more extensive consideration of the evaluation of dose response based on the mode of action key events framework that the HESI Research Foundation published a couple of years ago. Also, a couple of case studies, which you'll hear about this afternoon in brief, and approaches to cumulative risk assessment as visualized through the RISK21 matrix, or lens, I should say. I hope I've provided at least a little flavor of what RISK21 is about, but there will be lots of questions which we hope to be able to address later on this afternoon. But if issues come up in the days or weeks to come, please don't hesitate to contact one of the people involved with the project, and the key individuals are listed here with their email addresses, and we'll certainly get back to you as soon as possible. Thank you.
Wonderful, perfect timing. Thank you so much.
That's what, being a lecturer, you see.
And I think since we will be having a lot of opportunity for questions, unless there's a burning one right now, and learn more about the proposal here before, I guess, we take it apart. The one word you mentioned before was public. Yep. Which raised some eyebrows, and we may want to discuss the pros and cons available publicly. But anybody have any questions right now? Yeah.
Publicly accessible has to be.
Dr. Gager in Center for Tobacco Products. One of my questions goes to when you're referring the range of toxicity values and extrapolating that effect versus exposure. You're taking into effect a range of effects, but how are you taking into account the severity of the different effects that cross that range of effects that may range from inflammation to morbidity?
Right. So, of course, that's an issue in any risk assessment. There have been recent efforts to try to quantify that through the International Programme on Chemical Safety. And we would seek to incorporate a strategy developed elsewhere, which would, if one desired, weight the severity of the effects. I mean, one obvious way is to plot the toxicity values as a health metric like a QALY, which would take into account severity, duration, magnitude of effect. Of course, there are problems extrapolating from animals, let alone in vitro data. But there are methodologies which allow you to go from a point of departure for any endpoint, obviously, to a more sophisticated health metric. And so one could envisage ways of doing that, but it would need significant discussion, which we haven't gone into in detail in RISK21 yet, but it's certainly a very interesting point which we will explore. Yeah.
Just to keep on track, unless there's any really follow-up.
So waiting is,
right ahead. Oh. We'll welcome Dr. Michael DeLarco.
Thank you. I have. I'm more of a keyboard guy, so anyway, well, it's certainly delightful to be here today, and I want to thank our sponsors for their interest in RISK21. And my presentation today will be describing the activities of the exposure subteam, and has been referenced in the previous presentations. One of the unique features of RISK21 is the emphasis perhaps early on in the process of utilizing exposure data. So if we look at RISK21 as a framework, the objectives are to utilize the existing data that we have based on the problem at hand and using problem formulation as the grounding for the assessment, and then to compile the information tools that we have, both in terms of the array of toxicity tests that we know and love, to provide information that we can utilize to assess the degree of safety. And as was eloquently described in the initial presentation, safety is really what it's all about. One of the unique features of RISK21 is, can we take exposure data and in some capacity parallel the efforts that occur in toxic testing by somehow categorizing exposure tests and exposure data in a manner that would permit some type of a parallel exercise whereby, based on the amount of available data that we have and the interest that we have in terms of the degree of certainty that we want about the problem we're trying to assess, systematically arrive at a process of making a risk decision and then providing some degree of safety. And so then what the RISK21 is attempting to do is to, in a transparent manner, provide an opportunity to categorize and evaluate the existing data in a systematic fashion that provides an opportunity for the user to use only that data which really is essential to make the decision, and then in that particular capacity move up the chain as needed to provide additional data that is required for the safety assessment. And there are some benefits for relying on exposure data early. Certainly with the problem formulation, you quickly get into an arena where the exposure issue would guide both the amount and type of toxicity data necessary for a risk assessment by providing information about the route of exposure, persistence of the chemical in the environment. And one can imagine, as was shown in the last presentation, that if your exposure estimate is low, it's quite likely that the degree of toxicity information you would need could be correspondingly less, yet sufficient to make an acceptable level of risk and an estimate that would allow you to determine a safety level. And from a public health perspective, exposure assessments can certainly be used to prioritize chemicals for further study. By considering those chemicals that exist in the environment, persist in the environment, and have widespread usage, we can then make a determination of whether or not we have sufficient toxicity data for those chemicals, and then use the tools at our disposal to provide more information about the level of toxicity. And perhaps with this type of approach, we might not then be captive to the fancy of advocacy about a particular chemical of interest. So our exposure subteam then set out to categorize exposure data in the following manner by trying to group the various data in a way that could be somewhat meaningful in the risk assessment process. And again, we were somewhat informed by the process underway, as mentioned in the last presentation, about the rather explosive activities in the field of toxicology. And so the question now becomes for us is, can we somehow in some way mimic that approach? And so with our initial tier, what we call the minimal information, occurs in instances which we often face where we know very little information about the use of the chemical and much about its existence in the environment. We may have some limited information about its intended uses, some information about maybe its presence in the environment, but we really don't know much else other than physical chemical characteristics. And we want to know whether or not we might be able to utilize this information either directly or perhaps indirectly with structure-activity relationships to have some information about the exposure. We frequently encounter situations where we do have use-specific information and we can model the fate or persistence of a particular chemical in the environment in a deterministic manner. We frequently encounter that information and use models to provide more information about exposure characteristics. And likewise, with several types of measurements, we might be able to use probabilistic tools to provide somewhat of a distribution and range if we have sufficient data for input. And finally, ideally, we frequently have the luxury of biomonitoring data, which provides information about chemical contaminants in the body. And we can utilize that information directly to provide an estimate of dosimetry, or perhaps we can calculate with a variety of tools and techniques to provide some information about exposure. So to emphasize the roadmap, the real process that drives entry into this particular tiered structure is the problem formulation. By defining the problem and the nature of the assessment that we're interested in, it determines to what degree both you could enter the system here in terms of the amount of data you have, and also provide some gauge in terms of how much data you need. And so we hope that this particular approach would be somewhat of a buffer against the temptation to always continue to strive for yet more and more data to make a decision, when in fact, in some instances, you might actually have a sufficient amount of information, particularly in the area of exposure, that would allow you to proceed through the roadmap. So our next task was then, with the tier structure in place, was attempt to populate the various tiers with tools, models, and databases that could be utilized. And this is a process that's underway now in our group to begin to look at a variety of models and tools that are being applied in a variety of instances to determine whether or not they might fit this tier structure and whether or not existing data might be employed in some manner to begin to investigate how we might be able to rely and categorize existing data for risk assessment. Much of this data is available. The models and the documentation and support networks are available for models such as the USTOX model. And there's support being provided by several regulatory organizations. We also are finding that biomonitoring data is being made more widely available for a variety of toxicants. And so we feel that it's quite possible that we may have a rather large repertoire of tools and techniques available to support our tier structure. But again, this is a work in progress and it's still under evaluation. And in fact, we've spent the initial part of our effort with this tiered structure looking at the initial tier 0 and tier 1 levels, asking ourselves, is there a way that we might be able to use exposure data in a quick screening level manner? And our approach initially has been looking at banding-based models. There are exposure tools developed for the REACH program, for example, that provide information. There's also some generic risk and exposure assessment tools that are available that may indeed be useful for screening. They're intentionally conservative. They rely on this banding concept. And they're publicly available, which is very attractive in terms of access and availability and lack of proprietary information, which sometimes accompanies certain models. So among the systems are occupational exposure bands, whereby we look at various process categories and we can try to map to those categories. These bands are based largely on fugacity. They generally provide information about inhalation or dermal contact exposure. They deal largely with industrial workers, but oftentimes with professional workers. Likewise, there's a consumer exposure banding available for various products and articles. Again, the system of banding is based on vapor pressure. In these categories, we find the estimates for inhalation, dermal, and oral routes of exposure, spanning in acute usage or sometimes chronic usage, depending upon the nature of the product or the article. So for the remainder of my presentation, I'd like to illustrate how we're trying to take this approach further in terms of utilizing this kind of information to determine if indeed it might be helpful and useful to catalog exposure data and utilize it in some type of a structured way for risk assessment. So referring back then to our problem formulation in terms of the substance and what we'd like to know about it and what we know about it ahead of time, you know, we can then say, in this particular example, consider pyrethroid exposures where we're interested perhaps in bed nets that are dipped with pyrethroids for children who are 6 to 12-year-olds who would be the consumers or users of these nets. and also adult workers who would be involved in processing the product. And so the routes of exposure immediately, just from the nature of the problem formulation, would be inhalation and dermal. We really wouldn't have much of an interest or expect much of a concern from the oral route of exposure based on our scenario and our population of interest. And if we look to existing data, we'll see that the World Health Organization actually has a document about treating bed nets. So we have some information about the actual production process. And so the question is, can we somehow relate the problem formulation information to some of these existing data to provide some estimate of exposure? So if we look at physical chemical characteristics and we use delta methrin as our example chemical, we can get some information about the physical chemical properties and find that it has a low vapor pressure and it's not readily soluble in water. That then can provide us some information, for example, about the likelihood of having an environmental contribution, considering water solubility as our worst case estimate, and somewhat arriving at an indication that it's not likely to have a large environmental component. We can then concentrate our efforts on our worker and consumer scenarios, again, looking at the tables that are available in the lookup tools for industry and consumers. And then a third step later on would be to examine and compare some of our exposure assessments with various toxicological parameters. And so this gives you an example of the kind of lookup table tool that we rely on. This is for industrial workers. A variety of process categories are available and information about the dermal exposure and inhalation exposure. And so then what we're attempting to do in our exercise now is look to see whether or not there's a value added here by trying to align the exposures of interest to us with those that exist in the tables, and then be able to try to provide some type of a quantitative estimate both for the dermal contact and inhalation that can be utilized in the risk assessment roadmap of RISK21. Likewise, there's exposure information, again available in some of these lookup tools based largely on the type of article and then looking at the specific product subcategory. And in the case of bed nets, we can see that bedding and mattresses most closely approximates our particular situation. And again, based on the banding, we can obtain information about the likely exposure level. And then with some judgment about the nature of our product, we can further refine that to make a determination about the likely content of the treated product in terms of the situation that we're dealing with. And this can be further refined by going to the existing information, looking, for example, in more detail about the amount of time spent in transferring the chemicals from containers, the actual time workers spend in terms of the production process, the number of treatments at any particular plant, and the total number of nets that would be produced. All this could be estimated from some of these existing data sets, and we can then again go back and refer back to our scenarios. Also, we can get additional default information from existing guidelines, and we can compare this to toxicity estimates. So this is some of the work that we have underway. So we're on the way to establishing a proof of concept that exposure estimates can be made from existing data, looking initially at these lookup tables, trying to apply that information more widely for exposure assessment, but we really have much more work to do to verify the reliability of the process. We're focusing our areas primarily on these initial tiers where little data is available because we frequently encounter that in our work where we have chemicals of interest but not much exposure data. Yet it's important to realize that much more research is needed to determine if the higher tier structure where we do have data really will fit our categorization scheme and whether or not we can obtain some useful information from those particular tiers. The application of this approach would be enhanced by our ability to refine and standardize default values. In many instances, there's not much harmonization. Adding scenarios of interest would be quite valuable. There's really, again, quite a bit of variability in terms of how scenarios are constructed, many of them very generous. Certainly a consideration for weight fractions that would be oriented around consumer use, for example, when we're using data from the industrial arena would be helpful. And then perhaps investigating additional physical chemical properties and structure activity relationships might provide some insights, again, in those instances where we don't have much information. So I say that we're encouraged by our initial efforts. We have a long way to go, and we're proceeding ahead. We feel that we do have the opportunity to provide some additional structure on exposure data for risk analysis. Just want to acknowledge the sub-team members that you see here. They come from a variety of institutions, and they bring together collectively considerable expertise in the field of exposure analysis that we're applying to this particular effort. Thank you.
Again, thank you for being so timely and for giving an excellent further introduction to the topic. Is there any question right now? We have a little time for it right now. Dr. Collins?
One question. I've been involved in a certain metal, and I'm looking at exposure. Close. Okay. I've been involved in looking at a certain metal and looking at some of these lookup tables, and some of these values vary considerably. My concern is the accuracy in some of these tables.
Right. So there's a couple of points there. Our initial approach, first of all, we're in agreement that there could be questions about the accuracy. So one of the aspects that we're investigating is getting a better understanding of the basis of what goes into those particular values. It's very tempting to go to a lookup table and just grab the values. We've also done some exploration in terms of looking, when we do understand the nature of the contributing analyses that led to those tables, we've also done some work ourselves to do some additional calculations to see how close we come, let's say, to those particular values. And it's a challenge. We feel that we just need to get a better understanding of the inputs into these particular tables, and then also test the results that we get against cases where we have much better data sources to look to see whether or not this has a great degree of viability. But your point's well taken about being cautionary. And I'd have to say that when you look at some of these tables and databases and things like that, that background information is not always readily available.
Any other burning question right now?
So do you have any mechanism for worrying about how the duration of exposure matches up to exposure estimate? The type of exposures estimate you can vary a lot with. In some cases, there are different toxicity values.
Right, so that's going to be one of the more challenging areas, particularly when you think about the problem formulation and the scenario of interest and the interval of exposure or the time level.
Would you kindly just summarize the question? I didn't make it through the entirety of the room and probably not through the line.
So I believe the question was, let me just paraphrase, oriented around concern about differences that might be associated with the differences in exposure, that is, in terms of the differences that you're concerned about relative to the time intervals that one might find in particular existing data. Is that correct?
No. I think the point is you want to, the type of exposure assessment you do for acute exposure is going to differ from what you do for chronic exposure. And because the the toxicity can vary considerably. Sometimes it doesn't vary at all. For instance, acute and chronic are the same, and sometimes it makes much bigger with long-term exposure. And so just to get into it a little more, if you're worried about chronic exposure, you probably want a per capita intake estimate for a lifetime exposure. Whereas if you're worried about acute exposure, you're going to be worried about the upper, the short-term upper bound exposure. So it can vary a lot.
Right. No, that's we've encountered that particular difficulty in terms of mapping the exposure interval with the endpoint, and it's something we're aware of, and it really is not well developed yet in our system, but it will have to be accounted for, certainly.
Okay, thank you. How about we take a quick break? quick, meaning quick, as quick as you can, so that if we come back together and go through the next talks and we can start the panel as quicker, we are out of here. So how about 10 minutes, and I see you all back here. Thank you. Comes close to
Mic check, 1, 2, mic check.
Check 1, 2. Check something real fast.
Mic check, 1, 2, mic check.
Okay.
Mic check, 1, 2, mic check. Mic check, 1, 2.
All right. Maybe we can find our seats. Not working? Now? I think you really have to bite the microphone in order to make it work. All right. We had a little break and are ready for our next speaker. If we can find our seats and get back into our listening mode, slowly but surely. And we welcome our next speaker, who is Dr. Angelo Morato from the University of Milan. And I think your talk is already up.
Yes,
Are you good with the mouse and everything?
I hope.
Okay.
Oh, I can do that way. Good afternoon. Thank you for those who invited me to come here. It's always a challenge to be the only non-native speaker in the place. But anyway, I'll try to do my best to make me understood, both for language and for the meaning of what I will be saying. If you have seen something that is a little bit complicated before, I assure you that addressing the issue of cumulative risk is even more complicated, because you have different stressors or chemicals and non-chemical stressors to deal with, and as I will describe before. And one index of this complexity is that there is no uniform approach around the world on cumulative risk assessment. There are different regulations, there are different statutes, jurisdictions, that there are even different definitions of cumulative risk assessment. And therefore, there are different approaches. There are approaches that are restricted to group of chemicals based on mode of action. There are approaches that do cumulative risk assessment based on target organ. There are approaches that consider that chemical and non-chemical should be, stressors should be put together. And so really there is a need of trying to find out if a common framework on which to put in all these issues. Here is a partial summary of the many definitions that we have of cumulative risk, which I will not go into detail. Enough to say that, as I said before, you may have chemicals alone, chemicals defined in a specific way. You may have exposure to stressors other than chemicals included in the definition of cumulative risk assessment. So what we tried in RISK21 is to put all the information that come from the other groups regarding toxicology and exposure into a reasonable framework that make the complex problem of cumulative risk assessment manageable, one, and if manageable, then understandable and communicating this in a understandable way for the non-specialist. It's nothing, we didn't do nothing particularly new, because we started from what is already exists, mainly from the WHO IPCS framework, which is essentially a tiered approach for both exposure and toxicity in the analysis of the cumulative risk. So what we have done here essentially, we try to put into this framework the principle of RISK21, as has already been described before, and a consideration for non-chemical stressors. There are some definitions out there that say that you cannot call cumulative risk assessment if you don't consider non-chemical stressors, which it can be a reasonable position, but how can we make all these very complex issues of different chemicals and different non-chemical stressors manageable? How can we come up with an output which is useful, but an output which can be produced with the resources that we have at hand for the time presently. So what we wanted to do is to do a quantitative appraisal of the adverse health effect from combined exposure to multiple chemicals and non-chemical stressors. But upfront, we said, if we want to make this manageable, we should start from chemicals and then address the non-chemical stressors, which we named modulating factors, if and when necessary. But after we have dealt with the chemicals. So is a stepwise approach, and you will see how graphically how this will be, to manage this complex issue. It will be, as I show you, a process, an iterative process. You start with what you have, you assess whether what you have is enough to take a decision. If not, you go back, you reformulate your problem, and you refine your information on both toxicity and exposure, and you go on. What we meant by modulating factors. We meant all chemical and non-chemical stressors that go out of the initial group of chemicals that we want to assess. And these stressors may have effect on the toxicological outcome because they modify, for instance, the response of the individual, because they add a toxicological effect, an effect to the existing effect, because they modify exposure. And therefore, these may be different modulating factors, Dichter factors, belonging to the host, belonging to lifestyle, belonging to the environment. And one other thing that we try to do is to break down all these factors into a single factor to which we can, when possible, apply a number. For instance, the terms that vulnerability, susceptibility, and sensitivity, which always come up in all papers. They all have inbuilt a number of different factors. So what we wanted to do is to break down these into their individual components, be it individual susceptibility, be it changes in the toxic outcome, be it a change in the extent of exposure. So this is the driving concept for this. And for instance, this is a partial list that is still under development of all the modulating factors. As you see here, you should analyze them one by one. And you easily realize that some of these factors put together for some in the literature means vulnerability. For some means susceptibility. Well, what we wanted to do is simplify them. I always say I'm simple-minded. I need one piece at a time in order to make sense of all the information that we have. So that's what we try to do. And so the starting point will be the problem formulation for cumulative risk assessment. And the initial point of cumulative risk assessment, which is called a gatekeeper step, is, first of all, we need to identify whether there is evidence that indicates the need that the cumulative risk assessment needs to be carried out. And then you plan how you do it, you plan what you want to do, you consider other stressors, and I show you how you can do in a stepwise, and then you identify exposure effects, and then you do all the process. And So when a cumulative assessment is necessary, we need evidence that there is co-exposure and a common toxicity. We need to identify which compound need to go in what we call the common chemical assessment group. We can identify co-exposure with any of the models that have been described before. We can identify common toxicity or define common toxicity at different levels of refinement, depending on what we have in hand. We can start from starter-activity relationship until the most sophisticated definition of the mode of action. So starting, we have, again, these are the familiar triangle that you have seen, although not in this place in the slide. So we have evidence demonstrating co-exposure, different levels of refinement, or different decreasing level of uncertainty, if you wish, and the same for toxicity. So the question is, starting with exposure, do we have information that indicates that there is co-exposure? Obviously, if the question is no, then we just stop. There is no rationale to go on. If it's yes, then we need to ask ourselves, is there any evidence that there is a common ground for toxicity to group these substances and make a cumulative risk assessment? If the answer is no, then you stop. If the answer is yes, then you proceed. 2 points here. One, in this process, you see, you just need one no to stop your process. And the second point here is that although in some regulatory domains, in this case, I'm thinking mainly of Europe, there is a regulatory need to do risk assessment, cumulative risk assessment simply based on toxicity. Then, if you go to exposure, you may find a no there and stop there. So this system here is made in such a way that you can enter the system either from the left co-exposure or either from the right common toxicity. But you always have to ask both questions, and depending on the outcome of both questions, you may go on with the cumulative risk assessment. You may stop and not carry on the cumulative risk assessment. And our suggestion is to carry on this information for both co-exposure and common toxicity in tables, indicating what kind of evidence do you have to state that there is co-exposure, different levels, different tiers, different certainty or uncertainty around this information, and the same about the combined toxicity as a way of explaining what you are doing and also explaining to your mind what you are doing. So you have, in this way, evidence of both exposure and toxicity. And now you can start to do your exercise. So evidence of co-exposure and common toxicity, as I said, you just need a no and you can stop. Or you can go and formulate your problem. What is the problem that you are addressing? You identify your group of chemicals. You use the method, as I said, both for exposure assessment and toxicity assessment. And you use the plot. And I will show you how you can use the plot that you already saw to do your assessment. Then, based on the plot, you ask yourself, is the risk here is acceptable? If it's not, you start again. As I said, you can start any place on the levels that I showed you before regarding toxicity and exposure. It's not necessary to start from tier 0 if you have other data available. You can use the data that you are available. But in any case, if your answer is no, then that means that you need to refine And as Alan showed you before, based on what you see in your matrix, you may decide if you want to refine toxicity or you want to refine exposure information. If the answer to this is yes, then you ask yourself, are there modulating factors that modify the response or modify the exposure? Up to now, we have been discussing of an identified group of chemicals that you identified based on the criteria I showed you before. Now, the next question is, are there situations that modify the exposure or modify the toxic response or modify the internal dose that need to change or they need to be addressed and see how this is modified? So you replot your results based on the application of modulating factors, and you ask again the same question. If it is okay, then you stop there. If it is not, then you have some more work to do. Again, like, no, let's go. There are different ways of carrying on cumulative risk assessments. The other index and the cumulative risk index and the toxic equivalency, et cetera, et cetera. I'm not going into detail on this. I'm just showing you how you can use some of these in the matrix. This will be a little bit longer to explain. So here we have the well-known red, the green, so we have a concern here, and we have no concern here. Let's assume that based on the decision made before, this line represents the separation between the potential concern, the gray or yellow area, and the no concern. So in this case, what you have here, you have already these are the individual compounds, just a point estimate to make things simple because they are already complicated by themselves. In this case, you have these two compounds that are in the red area. And then you have a number of compounds in the green area. So already this will tell you that you don't need to go into the complex business of doing cumulative risk assessment until you solve the problem with these two chemicals. So just plotting this information will tell you that you have two problems to solve before you go into cumulative risk assessment. Another thing that this graph tells you is that probably, and this is, for instance, another line that you draw saying that if you are below this line, that means that these are so deep in the green that we don't need to put them in a cumulative risk assessment because they are so far away from any toxicological effect that they will add very little to any accumulation of effects. So you already from this, you have already some ideas of what to do next. So this goes away and these need to be solved. Fine. Now we solve them. These two have been solved because we did some more sophisticated analysis of exposure and or toxicity. And so they are below the red line. And we get rid of these because we don't, the exposure versus toxicity was so low that we didn't care about this. And so these are all the compounds that are here. You can then decide to use, for instance, the other index approach, identify an index compound, and express all the values, both in toxicity and exposure, normalized to the potency of each compound to 1 will be here. So you are in the yellow line, then you may decide if you want to refine it again to go back to a more green area, or you are satisfied with this. So let's assume that you are satisfied, and now you have a cumulative risk assessment for this group of compound, which is acceptable. Now we have the problem of the modulating factors. And again, We are suggesting that we identify each possible modulating factor, and see what the impact has on, if any, on exposure or on toxicity. The description of it, how strong it is, and which direction. Quantify if possible. You can do it for each chemical in the group, each modulating factor for each chemical in the group, or you may decide that you want to do it on the whole group, because there is no reason why one modulating factor will behave differently for one compound within the group than another compound. But in any case, if you want to evaluate for each compound, you can express the effects of the modulating factors with this arrow. So, for instance, in this case, to move here, this one moves here, this one moves here, etc., etc. And then you can redo the exercise that has been done before using applying the modulating factors. Or you can use it on the group, and then depending on the modulating factors, you may have different, the arrows in different directions. And then you will end up with another point somewhere here that will tell you whether there is concern or there is no concern. So this is where we are. I understand it is difficult. What we have tried to do is to have an approach which is manageable, feasible, which is transparent. Transparent not only to you who are doing this exercise, but to those who are reading or trying to understand your exercise. It is based on the problem formulation. It is driven by exposure. It is iterative, and you do how many iterations as needed to come to a result. The modulating factors, so all the external other factors are introduced later in the process when you have solved the problem with the individual group. And we think that it is transparent and visually should be rather simple. Also because all the steps are documented in a tabular form. And in this way, we also think that will be efficient in terms of resources, because this is an exercise that certainly will take a lot of efforts in terms of money and manpower. Thank you.
Thank you again for a very nice follow-up to what we've heard before and an in-depth view into this topic. So thank you very much. Are there any questions right now that we want to address specific to this talk, Jen?
Hi, does this approach take into account potential synergism like asbestos and smoking or aflatoxin and hepatitis, where the effect is more than additive and isn't predictable, really?
Well, one of the starting point of this exercise is that since the evidence so far available of deviation from additivity at human relevant exposures, and by this I mean exposure that are below the reference values for each individual compound, if this is occur, you do not expect significant deviation from those additivity. That means that synergy occurs when you have biological effect already there. But our situation is that you are below any detectable biological effect. That's why I said on the matrix said, when you have a dot in the red area, forget about cumulative risk assessment. You have to solve that one first. Because if you go back, you go to the green, yellow-green area, then the possibility of effects other than dose additivity is very low.
Question, Dr.
It seems like this is all focused on increasing toxicity, but what about interactions where there's a decrease in toxicity?
Sorry, I didn't.
It seems like all of this is predicated on a cumulative increase in toxicity.
Uh-huh.
But what about situations where there's actually a decrease in toxicity from cumulative exposure to different chemicals?
Well, the starting point is when you start the exercise is a group of chemicals that are supposed to act by the same mode of action. I mean, it's not correct. That are assumed to act by the same mode of action, okay? And then when you add other chemicals or other modulating factors that are non-chemicals, as I showed you in the matrix, there are arrows that goes towards reducing the toxicity toward adding to the toxicity. So this is taken into account. This is one point. The second point is that in the case of the work we are doing, if there is a reduction in toxicity, it's even better. It's not a problem, isn't it?
Okay, maybe we can use that for the discussion to tap on later. We go now into our last talk by Dr. Tim Pasteur, and we go from there to a short demonstration of the tool that we have the pleasure to be one of the first people to really experience today before we have our esteemed panel to discuss the burning questions on the forefront of all your minds. So hang in there, we are making good progress. And so right now we proceed. You found your talk? Is it up there? Yes.
It is.
There we go. Very good. You're good with the pointer?
Yeah. Good. Well, after a considerable amount of thought by over 120 people, as you saw from Dr. Bubis's presentation earlier, we had a lot of people coming up with these kinds of thoughts and the complicated nature of cumulative risk. How do you go about a very difficult subject of ascertaining exposure, which I think is probably on everyone's mind here when you do your day-to-day job. And of course, the whole tier-wise procedure of going through how do you get the best toxicological information that's relevant to human beings? A lot of thought went into this, and you're seeing that today. But what I'm finishing up with now is a question that we put to ourselves early on, which was to say what we need to do now is pressure test it. We need to take a look at taking those principles and actually applying it to some real-life types of problems and issues. And we came up with two scenarios, two case studies that I'm going to talk to you about. The first one is on pseudomethrin. The pseudomethrin, of course, is a pyrethroid, and it's a made-up one, which I'm going to explain to you exactly what it is. But we're going to carry you through the process of looking at that particular case study. It's a very data-rich situation. So we picked one where we had lots and lots of depth in the information that we could draw upon. The other example that we have is water contamination. And in that situation, we're looking at a lot of chemicals, but not necessarily having a lot of information. So how would RISK21 cope with these two examples that we're giving you today? So the first one that I'm going to go into is going to be pseudomethrin. And what I'd like to take you back to very briefly is the roadmap and how that leads us through making our decisions. And I've labeled them 1, 2, 3, and 4 to make it obvious, but it really is a paradigm where we're saying what RISK21 is all about is it's a problem formulation based, exposure driven, value of information systematic approach that's very transparent and presents that information in a very visual fashion so that you can see and decide to haggle, if you wish, over any bit of information you have here, and you know what you need to do as far as refining your information. So we use that in both of these case studies. So let me turn to pseudomethrin, problem formulation. What we decided to do is say, all right, you're a company, you want to put your pyrethroid onto bed netting, and you're thinking of having the local people actually dipping that netting into a bucket of this pseudomethrin. And you know that once it's dried and out there as a bed net, you have children and you have adults sleeping under that. And the question is that going to be safe? And the whole thing about RISK21 is to say you need to acquire enough information so that you can make a decision on safety. And you have to do so in a fashion that says, well, we're going to acquire enough information that has enough precision to make that decision. Nice little rhyme, but it's one that we go by, enough precision to make a decision. And in this situation, we're talking about another pyrethroid in a group of pyrethroids for which we have a lot of information. And the other thing that we're saying about this is that we need to determine reasonable certainty of no harm by using no more than 50 animals. So we're putting that problem formulation in front of the team to say, could you do this, given the information you have on the group of pyrethroids that are available? So we went through the process, we did the problem formulation. The next thing is it's exposure driven. So the question was, how much exposure will there be? And I should say also on this particular example, what I'll be doing is talking about sleeping under the bed nets. I won't get into, I'll mention very briefly about the bed net dipping, which was an unacceptable scenario. This one, in the interest of time, I'm going to go through just the sleeping under the bed nets. So the question was, how much exposure is there? And we start at the bottom of this inverted triangle because, as you know, as we go up the triangle, we're expending more resources, more time, and more effort to get more precise data. Let's start, first of all, with what we have in hand. And what we have in hand is actually a World Health Organization document that actually chronicles and goes into detail about how much exposure could occur to someone sleeping under a bed net that was impregnated with a compound. And the numbers are available. Now we know also, based on the physical chemical properties, now remember we're at tier 0, that this is a low volatility compound, inhalation is negligible. These are kids, infants, adults sleeping under these bed nets every night, so it's a chronic, maybe a subchronic duration of exposure. And we know that the World Health Organization model derives a value that is in a span of 0.001 to 0.443 milligrams per kilogram per day. And you can see it in this table, how it breaks out in the aggregate amount that would be exposed to adults, children, and infants by the oral and dermal routes. Now, the point here is that what WHO came up with was a very rough estimate based on it could be 2.5% dermal penetration all the way up to 100%. We're not sure what it's going to be, so what we'll do is we'll make the assumption that it could be anywhere in that range. So that's how they come up with such a large span in here that goes all the way up to 0.4 milligrams per kilogram per day. Now, what does that look like when we do the next step? And that's the visualization actually onto the matrix. And that number comes out as a fairly wide span. The precision there is obviously not that great. It's quite wide. Now, if the toxicity shows up as being fairly low, that might be sufficient. It might be enough precision to make a decision. So the next step, of course, is to go after the toxicity information. And in this example, what we're saying here is start at the bottom, see what you've got, and see if you have sufficient information on this new pyrethroid to make a decision. And the answer is fixed in the fact that there are all these other pyrethroids available to use to take data from. We know that it's a neurological endpoint that's most sensitive. We also know that there are structural similarities that are quite predictable across this class. Permethrin, for example, has the entire class has an acid moiety and an alcohol moiety and a central ester in the middle, which either has a cyano group or not on it. So you've got groups that are cyanide substituted over here or not. So we have two subgroups within the pyrethroids. Now, what do we know about the data? We can go in and do a read across and look at short-term, intermediate, and long-term no effect levels across all of these compounds here. And we can look at it with the cyano and the non-cyano groups, or the alpha cyano groups. And I can reveal at this point in time that our pseudomethrin is actually deltamethrin. It was the example that Mike used earlier today in talking about the example the exposure team went into. But we intentionally took this one out to see exactly how the data would actually come out if we were to actually derive it by the RISK21 procedures. Now, if you remember the problem formulation and information that we have already, we're saying this is going to be chronic exposure, so we're using values down in here. We're also saying that it is an alpha cyano substituted pyrethroid, so it belongs in this category over here. The range of toxicity information spans from 0.5 milligrams per kilogram per day up to 7.5. That's what's the range we have with this group of chemicals. Now, how would that look on a specific plot? Would be like this. The chronic no effect levels are in this range. Now, we have to bear in mind, number one, that the registered pyrethroids are the ones that succeeded. But there may be other ones out there that are more potent that didn't get registered. So we chose and documented in the paper we're writing on this that you could have, at this stage, the tier 0 assessment, you could have toxicity tenfold more potent than the range that we now have determined. So what we added on to that was a 10x adjustment factor plus the usual 100-fold adjustment factor to make these axes like for like. This is human exposure. This is human toxicity that we're estimating. This is animal toxicity, but what we're doing is estimating human toxicity, and that's why we have this rather large range. When you bring those together in this matrix, as we've designed it, you find that tier 0 does not give an assurance of safety, because if you draw a one-for-one line on the margin of exposure, you can see that it significantly exceeds that diagonal line. Exposure is wide, toxicity is quite expansive as well. So we need more precision. So the first thing we did is we went back to the WHO document and looked at the information available on exposure. And you remember I was talking to you about how much goes through the skin. It could be 2.5% to 100%. That's what's represented here on this particular graph. We went back and realized that it isn't 2.5 to 100%. It's actually a narrower range. Other assumptions that were in the WHO document that they documented as well as a more precise estimate of exposure allows us to actually make an estimate that is more precise. And you can see that represented here. You can also see that we still don't quite find that this is an acceptable scenario. So what do we do next? Class. Okay, we're going to go after the toxicity next. I was just seeing if you're awake out there. Okay, so the next thing we're going to do here is we have a 1,000-fold uncertainty factor or adjustment factor that we've put on this. Now that's reasonable given the fact that you may have, number one, a more potent pyrethroid out there than you previously knew about. And secondly, you have the 100-fold safety uncertainty factor that we typically put on. So let's talk about that 10x factor on the basis of something being perhaps more potent. We know a lot about the mode of action on these compounds. So value of information. We know that they have this series of adverse outcome pathway related events. And we also know that there are in vitro tests that we can do, non-animal based. And we can actually look at an in vitro test called the, and I know I'm going to get this wrong, but it's the microencephalographic assay, I believe it is. Which is a way of looking at the gate opening and closing. I know we've got a neurotoxicologist in here that knows this study here, and you can explain it in more detail. But this particular IC50 study gives us a range of values here that allows us to get some idea of whether our new pyrethroid is going to be within the range of what we already have registered, or whether it's going to be more potent or less potent. And you can see in this particular instance here that deltamethrin comes out at 175, in a range from 25 to 15-18. So you can see that we're within that range, so there's no reason to have that extra 10x factor in there. So what looks like the previous slide that I showed you as far as exposure here, now we're reducing these adjustment factors by 10x, and it's bringing it down into an acceptable range. So that's the second assessment that we've done. The first assessment was just general wild information that we could pull up, the WHO information, the tox information, we put that out. Second assessment, we have a refined exposure assessment, exposure estimate, and a refined toxicity estimate that gives us this visualization. So again, very transparent way of doing this. Now the third assessment was done by actually looking at using 24 animals to do toxicity studies that were targeted specifically for what we know happens with with pyrethroids, and that is neurological effects. We also looked at ToxCast assays to see if there were non-neurological effects that we could potentially see that we didn't see with the other pyrethroids, and didn't see anything there with alerts. So we reduced that by another 10x, giving us this visualization of the risk assessment for the pyrethroids. Now the bottom line on this I think is very interesting if you look at this table. Tox21, as Dr. Bubis was talking about early in his talk, was asking us for a jump shift. What Ron was talking about as well was getting a jump shift into another way of looking at things. Is there a different way of assessing toxicity, doing risk assessments, assessing exposure? And the answer is yes. And this is one way of going about it. The full regulatory studies package would tell you no more or less than the RISK21 stepwise approach on neurotoxicity or on the other effects. And indeed, the no effect level that we came up with from the 24 animal dog study is the same as what we had in reality for deltamethrin in the full regulatory studies package. But the real interesting thing here that I find is that this is something that is saving animals as well. And in this particular case, the full regulatory studies package is over 2,000 animals. And we can do this with 24 and utilizing in vitro assays and looking at ToxCast as well to get non-predictable effects. So this is an example, one example where you have a lot of data that you can draw upon and get a pretty good case study. The second example that we used for a case study was 133 chemicals in water, which we had to cut down to 20 because trying to get 133 chemicals on one of these graphs is very difficult, if not trying to get that kind of information pulled together in an efficient way. So we cut that down to 20 chemicals using the same paradigm, problem formulation based, exposure driven, value of information, and a very visual application so that you can see it. The problem formulation in this case was 20 chemicals were detected in surface and groundwater that may appear, that probably would appear in drinking water. Which ones, you have a year to decide which ones are of highest concern that we have to deal with. And you want to maximize your use of existing knowledge without issuing additional studies and animal studies and so forth, but you want to take advantage of existing knowledge. So in Tier 0, what we're looking at is using worst case exposure estimates and comparing it to the TTC, the threshold for toxicological concern. The next tier, or what we're doing in that situation, is using a Tier 1 exposure model, like Mike was talking about, using solubility, maximum solubility as a way of getting at how much would be in water and a maximum exposure, and again, comparing that to the TTC. And finally, with Tier 2, what we're doing is utilizing that same Tier 1 exposure model and looking at existing chronic tox values. So that's the methodology that we used in this particular case study, which of course is very broad. This is the list of chemicals, 20 chemicals that we looked at, and in this particular case you can see the Kramer class for each, the TTC values. And when you visualize that, now I need to explain here, this is still the matrix, but for those of you familiar with TTC, there is no toxicity component to that. It's an exposure-based paradigm. So you have on here estimates of exposure running from very low to very high. You have these vertical lines that represent Kramer class 1, Kramer class 2, the cutoff for OPs and carbamates, and then gene tox, structural alerts for gene tox. So that's the same matrix that we've shown you before, but now it's in a TTC context. Now what happens when you put these vertical dotted lines in for the 20 chemicals? You see them all coming through here, and you can see that 2 chemicals of the 20 will drop out because they're below the level of concern based on TTC. So now we're down to 18 chemicals from 20. What about those 18 chemicals? If you then refine the exposure value by using water solubility, and I believe I said the tier 1, what you find in this situation is you have now refined these vertical lines, which are your individual 18 chemicals, and you end up eliminating an additional 11 chemicals that are below the threshold for toxicological concern. 20 minus 11, we're getting down to just a few chemicals here. This is 11 out of the 18, by the way. So we're down to 7 chemicals. The next thing we do is we've gone around here. Now we go to the toxicological information and say, what do we have that's available that will inform us on those remaining 7 chemicals? On those 7 chemicals, we have exposure models, certainly we can use here, but we have toxicity values that we can now employ in using the RISK21 matrix. And we visualize it with this diagonal line, and we can see here very clearly which ones, perhaps these 2 that are still of concern, other ones that are not of concern. So again, enough precision to make a decision. And we've answered the problem formulation in this situation, and we said these are now 7 chemicals that we may be concerned about. And depending on your situation and the regulatory context, you may decide to pinpoint these 2 and leave these other ones alone, or you may take on all 7 of these. But at least you have gone through the process in a transparent way and presented it in a very visual fashion. So by using the TTC and this exposure information, we could winnow this down from 20 all the way down to 7. And we haven't done a single toxicity study. This is Tox 21 at work. The next steps we have to do is you can gather more information, you can plot the ranges when you need to, and use available information to the next iteration of the RISK21 roadmap. So now you've seen all the way from Dr. Bubas's presentation at the beginning of the thought process by over 120 people over the last several years to come up with some thoughtful ways of going about the risk assessment paradigm. Is it earth-shakingly different? I'll leave that for you to decide. Is it something that would make a more transparent and obvious way of going about risk assessment? I really do think so. I think it's a way of going about things in the 21st century that'll make a difference in the risk assessments that we do. Now, one of the best parts about this, as we got to thinking about how this is a process that we think is very, very usable. We started thinking that we could give talks like this till the cows come home. And indeed, we have done that in Japan and South Korea, have been in a lot of different places talking about this. And we've generated a lot of excitement, a lot of great questions and feedback that have helped develop what we have now. But we also decided that one of the best ways to get people to start experimenting with this and get it into more common practice is actually develop a web tool that each of one of you could use that gives that transparency, problem formulation based, exposure driven transparency and value of information that gives you a risk assessment that is easily explainable. I understand that there's a risk communicator out there somewhere. I hope that person is still here. Yeah. And one of the things that we all have to do is communicate what it means when you get a chemical spill in a West Virginia drinking water supply. It means telling somebody about what dredging a river is going to mean when you are starting to bring up metals into the water column. It has to do with what you do in communicating that information to people about what packaging is going around their food. So all of those things are what RISK21 can have an impact on. And what we're looking for today is some feedback from you in this panel that'll give us an opportunity to take it to the next stage. As Alan pointed out, we've got 10 publications that are coming out in the next 3 to 6, 7 months. We may have more to articulate this, but one of the most exciting parts, and ladies and gentlemen, the first on the planet, no one has seen this done before. We're going to show you the web tool. And Michelle tells me that she's not going to break it. But if she does, it's only because it's an alpha version. We've only played around with it since this weekend. We got it, what, Saturday? Saturday. And we started playing around with it a little bit, and Michelle is going to walk you through how to do this.
And then you can take some questions, too. Oop, I didn't mean to do that.
So do you want the questions now, or you want them after you do the presentation?
While she's getting set up, why don't you, if there's any
Okay.
questions
Any burning questions
on the case
related
studies?
to the examples that we just saw?
Thank you very much. That's a very nice presentation. One of the bits of feedback that I would like to give to anybody engaged in this kind of an approach is you're making an awful lot of assumptions about the correspondence of toxicities to structures. And so whether this kind of approach is acceptable or not, to me, depends upon whether how important the decision, the regulatory decision is, how severe the toxic effects are. Now, I know with, I think the pyrethrins are what they also call neonicotinoids.
No, it's a different class.
Is there a different class? But I know there, with that class of insecticides or pesticides, that they're starting to suspect that it has effects regarding the bee collapse or colony collapse syndrome. And my point is there can be other toxicities to other systems that are not necessarily characterized by the tool set that you have demonstrated.
Right.
And that's something that, you know, it extends to the whole animal system versus using organ representations of organ responses. How do those individual organ responses, how are they altered by their interaction within the whole organism?
Yeah.
It's another question that I feel is not really addressed yet by this kind of an approach.
I agree with everything you're saying as far as the reliability of QSAR has been questioned over time. No doubt about that. That's why we built into this the opportunity to look at these pyrethroids through the ToxCast assays. For looking at adverse effects that are not neurological. Now, how reliable is that is really the question. Maybe, maybe not.
You said that this particular material that you used as your test case, that it was data rich. What did its own database say about toxicity in other organ systems? What about using that information and comparing it to what the RISK21 prediction would be?
Yeah, and in the paper that we're writing, we do that. We make the comparison. We look at other adverse effects by the pyrethroids and look at specific ToxCast assays that would predict or not predict that, and anything else that might trigger some suspicion that that particular structure is going to be an outlier from what we already know in that structural class. You know, one of the most important parts about the thinking that we had in all of RISK21 is this tier-wise approach. And so at the bottom of that inverted triangle, you're talking about a tier 0, make the best of whatever you've got. And then you move to the next layer if you need more precision to make that decision. So in the case of a dread disease, for example, you would not take that lightly and just make some decision about something without having more information if you needed that. But if, like on the chart that you see here, your exposures are extraordinarily low, you would have to assume that your toxicity would have to be extraordinarily high to be relevant to human health. And that's what we're trying to get at, is that you want to find the information that's precise enough to make a solid decision about human safety. And you can't necessarily, if you're not comfortable at that particular level on that inverted triangle, you have to move to the next level. But this is a way of visualizing your preconceived notion of what that is. Any other questions? Yeah, in the back.
Yes, hi. I just have a quick clarification question about what you're showing on the map. Is that an estimate of risk to an individual or in a population of 100,000 people? Or is it simply a mapping of the two metrics where the values are?
It's simply a mapping of where the two values are with the incorporation of adjustment factors. So in that sense, it's a traditional risk assessment.
So maybe we should go ahead and see the tool now, and then keep the questions to engage our panel and have it a little bit more of a back and forth. So this is Michelle Embry from HESI. She will walk you through this brand new off the press.
Yeah, which I'm learning myself, so patience. We just, as Tim had said, I think we got the actual link for this just this weekend. But one of the things you've all seen today through the presentations is this plotting and the idea that you can really, in a lot of ways, when you're working on a risk assessment, easily visualize where you are. And then, I think Tim asked that question, where do you need to get more information? Where do you need to go? How might you apply this in scenarios like cumulative risk, et cetera? So we've been working with this company called Psyome to try to get this pulled together. And as Alan Bubis said, this is really an alpha version. Eventually, the idea is that rather than just being simple user input, is that you could maybe link to databases, or you could link to models. You could put in a SMILES code and it would pull information for you. So this is the very, very most basic version. We always joke all those plots you saw on the slides today were generated in PowerPoint, which can get a bit tedious, especially when you're plotting a bunch of different values. So it's an alpha version. The plan really is to try to get this to the point probably over the next few months where we would be able to share the URL as the beta version, have people try it out, see what works. Just in the couple hours I've spent with it, there's probably 5 or 6 things that we've kind of said, we should add this in. I took some notes today on some things we could do. But one of the basic things here, the first thing I wanted to show you is and now it's not clicking. Sarah Webb. I knew this would happen. Hold on. Yeah, it's live. It's just not, maybe you need to refresh. It'll probably erase all the stuff I have in there. Nope. Oh, it did, but that's okay. No problem. I can make up numbers. So one of the things here, and what you've heard a lot about today, is the need to discuss the problem formulation. And I think this gets at maybe your question about the neonics and the bees, because what we've really been doing in RISK21 is looking at the application for human health. But obviously, if you had a question related to ecological risk assessment and you wanted to assess the effect of a particular chemical on the environment or a particular species, you would identify that in this problem formulation phase. And the reason we have that in there, and it's just a blank text box for now, is that none of this should be taken in isolation. So if you create a plot, you create a data table, you have to have the context to go with it, or it can be misinterpreted and misused and honestly isn't going to be very useful at all. So that's one of the things that I think we'll begin to refine is what we need, what is sort of a required field in problem formulation. And in a way, we might make it that it won't draw the map unless you put something in here, because I think it's very important. So just the basics here, you can change the title. The units, as we've talked about, for exposure and toxicity have to be the same on each axis, but you can change them. We have a few options here. We'll probably add in a user input as well. So you could change it to parts per million, parts per billion, mgs per kg, mgs per kg per day, whatever you'd like. You could identify what your minimum and max exposure and tox axes are depending on the data you have. So you don't have points that are plotted off the matrix, which often happened when we were trying this out. And then you can identify, we have this red zone and this green zone here, and this yellow area, and you can identify what your acceptable margin is. So it's 1-to-1 right now, but you could make it 1-to-10 or 1-to-100, and it'll automatically shift that line. So then based on your problem formulation and your context, whether it's prioritization, definitive risk assessment, whatever you're using it for, you can change that. So the rest of it's pretty basic. You can put in a chemical name. So you guys can all watch my typing, which will be always fun with an audience. You can choose your favorite color. And I actually was taking notes while Tim was talking. So for exposure, you can just right now, eventually you'd like to, of course, be able to link in to, say, probability distributions, or potentially copy and paste from an Excel spreadsheet where maybe you have 50 values and you could just copy and paste them in. But for now, it's just set up to give kind of a range. So say you had I think Tim had 0.0001, 2.4. For the pseudomethrin, you have an option to put a point estimate in if you have it. For the tox estimate, I think what you can do is you can either put a range or you could just put a point estimate. I think you originally had a point estimate of 0.5 with an uncertainty factor of 1,000. That's 10,000, that's not good. That would be crazy. And then you just click OK, and it draws your box. And I spelled it acetaminophen, but that's okay. It's a new one. So what you see here, this little triangle, is the point estimate for tox, and it's applied this 1,000-fold safety factor. And then the exposure range that Tim had, or I made up, is here. And again, this is with a margin of exposure of 10. You could change that to 100, and it'll shift it down. What you can do then is you could actually add a new chemical if you wanted to do a refinement, or you could say, I think this should work. We'll see how good these computer programmers were. Change this to 100. It didn't. I think I have to add a new chemical. That's a feature that's in progress. Sorry about that. But basically the idea is what you could do is put that same data in. Bear with me here. And my point estimate's 0.5, but now I want to have my uncertainty factor of 100. In the end, what you would have, it's going to draw that box and shift that uncertainty factor down. And then what you could do is you could refine this exposure estimate, make that 0.01 or something, change it, and it'll show it and change it in real time. So the idea that those of us who were working on this project, is that you could visualize this very easily. You could get a handle on, as these numbers change, as you go through the different tiers, how does your picture change? What are the various factors you would want to modify? Eventually, one of the other things, I think the last thing I'll cover, and you could add as many chemicals as you wanted, add chemical, delete the chemical, modify. This would have a couple buttons up here that would create a picture of the sample plot for you, would create a data table of the data that went into the plot, and then also print out that all-important problem formulation piece. And again, the Zeta version will hopefully then link into data sources and make it really easy so that you're able to put information in. And again, a lot of it's a lot about transparency, communication, and things like that. And it's not something that we're not sharing the URL yet, but I'm hoping maybe by SOT time, we might be able to share it and have people just, try it, kick the tires a bit. And I think one of the things that we're hoping to get out of the panel discussion, which we'll start in a minute, is to get feedback from you guys here on how this might work. What things, if you played around with this web tool, would you want to see? What's useful, what's not useful, what makes sense, what doesn't? So I think we're at the point of the project, we'd really like to get a lot of feedback and appreciate everybody coming today to hear about it.
Great.
So.
Well, thank you.
So at this point, I think I want to ask the panelists to find their spot up at the podiums in front there. There are 6 chairs. There's water, there are pens if you want to make notes. And Dr. Susie Fitzpatrick will facilitate the discussion. And I will run around with the microphone.
And I also wanted to add, Craig Rowlands was unable to make it today because of the snow, but Alan Bubis has agreed to step in on the panel for him. Are there enough seats?
Sabine, are you sure we have enough water?
See, I want to make sure nobody dehydrates on my watch. Thanks for coming.
First of all, we don't get furloughed. It's called firing.
Right. It's a different world, but the goals are a lot clearer.
Yes.
So why don't we have all of our panel members introduce themselves, and then we can start with some questions.
You've seen me before, Alan Bubis from Imperial College London, an academic, but co-chair of the RISK21 steering group with Tim Pasteur.
Hi, I'm Doug Karras. I work in communications here at FDA, more specifically for CORE, but I've worked on the FDA press desk and on press desks and in media relations in a lot of different organizations.
I'm Clark Carrington. I'm a toxicologist on chemical hazard assessment team, and we mainly work on contaminants.
Thank you, sir. You're very hard to understand, though. Could you maybe
Clark Carrington.
get that closer to you?
Is that better? Clark Carrington, and I work on the chemical hazard assessment team, which is primarily concerned with contaminants, which is, as I'll explain later, are unintentional.
Good afternoon. I'm Dennis Keefe. I'm the director of the Office of Food Additive Safety. I'm more of a risk manager than a risk assessor, so I think I might have a different perspective on this.
David Jacobson Cram. I'm the Associate Director of Pharmacology and Toxicology in the Office of New Drugs for the next 6 weeks. And,
and after that?
Then I'm just me.
Fishing?
I'm Doug Wolf. Since this last November, I'm the lead for toxicology in North America for Syngenta crop protection. But prior to that, I was 16 years with the US EPA in various roles, both in the research as well as regulatory. And you all understand the message of I don't speak for the agency. So now I actually don't speak for the agency.
Now you can do it.
No, I can't. I can speak about it, though.
So why don't we start with some questions from Michelle, and then we'll open it up.
Yeah, I think the purpose of the panel was really to just get some discussion going, and we had a list of questions, but the first one, and we're not going to go down the row, so just would invite the panelists to speak up, and then anyone in the audience with questions is really to get some feedback on how you would see the RISK21 approach being applied to the area of work that you're in. And obviously, Doug and Alan are on the RISK21 group, but they're also, they do have full-time jobs doing other things. So would invite them to weigh in as well. But just wanted to get some general perspectives from the folks on the panel. We have a diverse group here on their general thoughts. And then there's, if we have some more specific questions later. But would like to have this be less of a formal panel and have kind of it be an interaction between the audience and the folks sitting up here. So anyone want to take a crack at that one?
I'll start. So I think I've been part of this effort since its very inception, and so it's very exciting for me to start seeing this phase of the RISK21. And so most of the time I was at the US EPA as part of this, and over the course of the past year and a half, before I left the agency, we did have many opportunities to present this internally, and I presented the various stages of RISK21 within the Office of Pesticide Program, within Office of Water and various other, and we've presented it to the Office of the Science Advisor. And so there's a lot of interest within the EPA. Now, present two very specific scenarios quickly. One is in the Office of Water. If you're familiar with the candidate contaminant list of what drinking water contaminants should be regulated, I think you could quickly see how this type of approach, when you have a universe of 6,000 chemicals, how do you decide which one of them should filter to the top? And that's their universe they start with, of which ones do you need to make regulatory decisions, which ones do you have any exposure or any effects information on, and what do you need? And so there's a lot of interest in using this kind of approach already. So that's one area. And the other area, if you've been following the endocrine disruptor screening program, is they've made great strides. And I think you saw with the various presentations of using inherent properties, the physical chemical structure, what you know about use and exposure. And in the recent, last January, when they did a science advisory panel review of the EDSP, the initial cutoff was using physical chemical properties to get, and their use to get the original universe of over 10,000 chemicals to be tested through the endocrine disruptor screening program. At $1 million a chemical would be quite expensive. To down to the, after looking just at use and physical chemical properties, down to less than 4,500. So while that's still a lot, that's quite a bit less than 10,000 and a place to start. And from there, the next iteration will be using exposure and in silico models to whittle that down even more. So a lot of these are already being applied, and there's real opportunities to expand those roles within regulatory science.
I mean, in a different context, working with supranational advisory groups in two areas, I could think of really value in the application of this approach. One was we were faced at EFSA with a problem that some of you here will probably have been faced with, is the issue of emerging brominated flame retardants, BFRs. There's a whole bunch of these compounds which we're finding may be getting into the food supply, may potentially get into the food supply. There's not a lot of information on exposure, and there's even less on toxicity. And we were thinking, do we just write to the risk managers and say we know nothing? There's 50 of these compounds, do what you want. Or do we try to find a way of prioritizing them based on the limited information available? And I think the RISK21 approach would have allowed us to array the available information, recognizing there's a lot of uncertainty, but nevertheless to at least rank them in terms of where do you start and then where do you move down to. The second is another area which many may be facing, possibly not here, but certainly in terms of residues in food, which is the environmental degradation products and metabolites of pesticides. So these are substances to which we've never exposed a mammalian species to because they're not formed in the rodent or dog toxicity tests. They're formed uniquely in plants or in the environment. So the chemicals haven't been synthesized in the laboratory, and we need to find out whether human exposure is a problem. We've come up with a framework at EFSA, but I think the RISK21 approach would have provided invaluable transparency in arraying the information in a clear way of to which of these metabolites are the ones you really need to think about moving forward with a higher tier assessment, and where are the ones which are really deep in the green and you really just don't need to worry about. So I think that there's a lot of value in the approach in different risk assessment scenarios. The one thing I do want to emphasize is RISK21 is not toxicity 21. It's not about developing and evaluating new methods of toxicity testing. So the thing that underlies everything we're doing is if we're going to apply a QSAR or an in vitro test, somebody somewhere has to have established its fitness for purpose. So that's intrinsic to what we're doing. So in other words, we're not validating tests or assessment methods. We're assuming that they are useful for acquiring information. And of course, one of the big question marks out there at the present time is, what does the ToxCast assays tell us about the in vivo toxicity of these compounds? But we're not in the business of saying what they tell us. We're saying that when somebody eventually works that out, we can feed that in to the RISK21 approach. So that's an important distinction I wanted to make.
This is Dennis. Alan, thank you for making that point, because it
Again, more into the mic.
Hi there. Thank you for making that point. You know, my office is responsible for the pre-market review of submissions. So unlike, I think, Clark and others who are at EPA, where you're dealing with environmental contaminants, we have sort of the stick to say, look, give us this study, address this uncertainty. So using a tool like this, it's all very interesting, but you know, how does it fit into our decision making? And the other thing that struck me in the presentation today was, okay, in my office, we're constantly being bombarded by questions from the media about substance X that's in our books, that's approved. Why are you continuing to let this be used? And it's difficult enough to explain our risk assessments. What you put up on the board today would be even more difficult to explain to the general public who have never really thought about risk assessments and you know the continuum of risks that we have to deal with in making a safety decision. So I just throw that out for consideration by the group. Some of the challenges for us as regulators who have to make the decisions that an affirmative decision that yes, you know, this is safe to add to the food. It's not just getting into the food because of some environmental contaminant or some agricultural practice. It's intentionally in the food, and you're intended to consume this.
All right, let me follow that up. So I guess first I just would like to make a general comment about, you know, the idea of problem formulation. And I think, you know, if you work in a regulatory agency, a lot of times the problem formulation is done for you. So it's like, you know, it's already, you know, what the problem is you know, it comes in the door. And so like for additives and even for us most of the time, we know what the chemical is because that's what they're asking about. So, you know, so going out and trying to figure out what's the problem is just not part of the, you know, of what we do. And the other thing about, you know, especially in contaminants is I'd say most of the time if we're asked to evaluate it, we already know it's sort of like in the orange zone. It's like, so we're trying to figure out whether it's, you know, yellow or red. So so then it's usually a, you know, a close call. So, I mean, I guess.
So I do want to respond. So it obviously depends on the authority that you're working in as to how well defined the problem is. But I mean, I've certainly been asked to give advice to authorities where the problem has not been well formulated until there was a dialogue to allow the further better clarification of the problem. But I do accept problem formulation is not part of risk assessment. It sits above it. It's the interface between risk management and risk assessment. That's absolutely clear. And you're right. Sometimes the problem is sets itself. It's a new food additive. Is exposure in the food acceptable? I mean, that's a fairly obvious problem. In terms of the exposure, though, I mean, you must see we're being asked questions about contaminants across the whole range of exposures. Some are, I mean, literally somewhere like the margin of exposure was 1 millionfold below anything that was near a concern. And yet there was a risk. There was a risk. These are some of the really rare chemicals which are being used in a few industrial processes where the exposure is trivial. Yet there was a real risk that the public purse was going to be asked to spend large amounts of resource to pursue the risk assessment at a high tier. There was absolutely no need for that. And other ones, you're right, that they're really up in the gray zone where we really need to think about a higher tier assessment to know if there's a problem or not. But I think there is, at least in some authorities, a very wide range of questions about where we are across that matrix.
So I'd just like to comment. I assume I'm here because some discussion how this applies in drug development. So I have a new challenge actually for the group, because we need not just risk 21, we need risk benefit 21. And the truth is, you know, we're just scratching the surface on risk 21. Risk benefit 21, we don't have a clue how to do that. And that's why we had advisory committees, and they vote and they figure out, you know, when the risk and the benefit is worth it. But obviously, it's very, very subjective the way people look at relative risks and benefits. Now, there are exceptions, though. For example, phase 1 clinical trials are almost always done in healthy volunteers. In that case, there's only a risk assessment because a healthy volunteer has nothing to gain from that potential drug except monetary reimbursement. The other one has to do with impurities in drugs. The impurities themselves don't confer any benefit, and yet we have to do a risk assessment on them. But then when I hear about the cumulative risk, it's a really difficult thing to do. We don't try to figure out what the cumulative risk is of taking 2 pills or 3 pills. Those of us who are nearing retirement, none of us take 1 pill, right? I mean, I have to swallow like 6 times every morning. And it's always a different mix. So how do you figure out what that cumulative risk assessment is going to be? It's really challenging. I think I really enjoyed Tim's presentation. I think, you know, these tools are great, but how often, like with the pyrethrins, are we going to have 11 congeners in a data-rich environment? And we never see an 11th in class drug. You would never develop that. There's no money in it. And so these, if you're looking at a complex molecule, the chance that you're going to have a data-rich environment from so many others that came before it, it's going to be relatively rare, unfortunately.
Well, isn't that what Tox 21 is supposed to tell us? What nth in class is it? In other words, we're supposed to find commonality through biological pathways, which we haven't got yet. But the idea is there's not going to be 70,000 different pathways to toxicity. There's going to be a smaller number. I'm not going to dare give a number. But with the, I mean, and I'm not saying that high throughput is the answer either. But I'm saying that as we improve our knowledge of modes and mechanisms of action, we will understand more about the commonalities of the signaling pathways and the biological processes leading to adverse outcomes, and that will allow us to group compounds better, maybe on, for example, a biological profile of a new complex molecule, which tells us, well, actually, it fits with a biological space that we're familiar with, even although chemically it's different from most of the things we've seen before. I think that's the hope, expectation, and reasonable evidence that we have now, that one day, and I don't know how long, we're going to get to that position. And then that will allow much more application of these approaches.
Is this working? Okay, yeah. My quantum mechanics professor as an undergraduate claimed to be able to visualize problems in 8 dimensions. Unfortunately, I can't, and so there's going to be a struggle to think what the 3rd and 4th dimension on your plot is going to be. And so the one that I'd most like to know about is, I mean, I know what exposure is. It's usually one thing, but toxicity is many different things. And so where you draw the line, what's red, what's green, and what's yellow, and how broad that yellow band is, really depends on what the toxicity is. And when you know what the toxicity is, if you have an endpoint and you're trying to determine an acceptable exposure for that particular endpoint, that's a relatively easy question to answer. But when you have an unknown and you want to know what are the toxicities that I have to protect against, I don't quite understand how you figure out where the margin of exposure that you put is going to be on your graph.
I think that there's a slight misunderstanding of what the matrix is about, because at this most basic, it can simply array conventional toxicity data, and then you're faced with exactly the same questions we have now, only they're visually presented In other words, how do we interpret the different endpoints with respect to severity, species, duration of exposure? Then we start to apply alternative non-animal method data where we're going to be faced with exactly the same problems when we try to apply those methods in a risk assessment without using the matrix. We've got to think about, what does this in vitro test tell us? I think I saw an approach that's being pushed by the WHO, and it will be published, I suspect, as an environmental health criterion document soon, which is expressed, let me get this right, there are 3 dimensions to this. One is it's the 95% confidence interval on the percentage of a given percentage of the population suffering from a given percentage of a response. So it's like a 5% change in liver weight in 1% of the population. What's my 95% confidence limit on that estimate? They are proposing to communicate this to the public. And so there are approaches which we can use. As I've said, there are these multidimensional health metrics that in risk benefit, we haven't even started to use them properly. You can add in other dimensions to them. You can think about indirect health consequences, risks and benefits, and then factor them into this common currency, as we called it. So I think we have tried to develop a strategy, an approach, a roadmap, a tool, but I think that the way it's used is open to a lot of further development. There are multiplicity of ways, and we shouldn't be constrained by what we've shown. This is just the tip of the iceberg. I think the problems you've posed are very real, but I think that there are ways that once we start to think about them, we'll begin to array them on our matrix, we'll begin to get to grips with them. I can think of ways of presenting multidimensional severity data on that graph. For example, you could color code the severity by plotting each endpoint on the graph. And I would emphasize again, the conclusions about what you do about the position on the matrix the position on the matrix is the start of the discussion, not the end. It's the risk assessors then sit down amongst themselves and, depending upon the problem they're faced with, have to discuss, what do we do now? What's this telling us? You don't just say, well, we're in the yellow zone, therefore this is the answer. It very much depends upon what's the uncertainty about the estimates, what's the scope for refinement, what's the severity of the endpoint, and what's the purpose we're using this substance for.
Yeah, I had a comment going back to the previous one on the cumulative. I think that we should be always mindful of there are 2 levels of cumulative assessment. One is for substances that are provided to humans to have a biological effect like drugs. And the other one is for substances that enter the body because they are there, not because we want them to enter the body. And so the situation is, in one case you want to have biological effects, in the other case you don't want biological effects. And the situation for cumulative risk assessment is quite different, because in the first case you have biological effects, and the combination of 2 or more drugs in most of the cases cannot be predicted. It can be anywhere from reduction of activity to synergy to potentiation to antagonism to nothing. And so you need to address the problem for those compounds. In the other case, when we are exposed to substances that are not supposed, or we don't want them to enter the body, but they enter because they are there. As I said in my presentation, first, you have to solve the problem with the individual compounds that are above their reference values. And then you start dealing with cumulative risk assessment. And in that case, the evidence that we have available is that there are no significant deviation for those additivity, if there is those additivity. There is no synergy potentiation, etc. So we need to have this clear in mind.
I was wondering whether anybody as in the 120 scientists done
using this tool to apply to the food chemicals, food-related chemicals, food substances, food ingredients? And now the second question is, sounds like it could be a very useful tool, could be useful tool to as a characterizing the risk, how to put together information and explain it to the public. And
Yeah, we haven't
I'd like
specifically looked at food additives or food chemicals, but I think you point out that it is a tool that allows you to frame the conversation, and I think that's something. When we were first going through this, we had to keep pulling ourselves back and saying, okay, we're not trying to do a risk assessment using this. What we're trying to do is use this to stepwise have a conversation about, do we have enough information yet? And then if we don't, what's the additional information? And this is consistent both with in the Office of Pesticide Programs, where they can request data. They have an integrated approach to testing and assessment, where they're trying to pare down over time what information they actually need to make a risk assessment on a new crop protection product or for re-registration. And this kind of tool can really help you say, well, would better exposure data be all we need? Do we actually need that multi-gen study, or do we have enough information to make the decision? Do we need no more information for this re-registration? And it's very transparent. Now that I'm in a company, we are using this problem formulation approach within Syngenta to have a conversation about, well, if we had our druthers, what data do we need to produce? What kind of mode of action description do we need in order to move this product forward to the next level? And if we had that information, would it matter? And so while again, the tool is there to help you frame the conversation to get to the next paragraph. It's not to get you to the end of the book.
I think just regarding the question on the case study, we haven't done any more than what you saw today, but that's something that in pulling this together with Susie and Sabine, we talked about. What are some case examples that if we did, I know was mentioned at the beginning, if there was a workshop that was held later this year, are there some examples that you folks at CIFSAN think might be useful to try out? And I know there were a couple that were thrown out there, so I think that's something to consider. And if people have suggestions, I think we'd love to have, again, like I said, people try this out and kick the tires a bit.
I can certainly see the value of this kind of an approach when you have a very large number of compounds to try to deal with. You want to be able to do something to rank them by priority for further testing. This seems like an extremely valuable tool for that purpose. But let me take you to the other side of the spectrum, the toxicological spectrum, where a regulatory decision has to be made. In that case, the level of uncertainty that we are allowed to tolerate is relatively small. And if we overstep that bounds, in other words, if we don't use sufficient certainty to determine the decision, then non-government organizations and or specific professors within universities are going to let us know that. So I guess as far as I can see personally, what I think needs to happen is there needs to be some kind of correlation between what the task is, how far into the assessment process, risk or safety process, you actually have to go in order to supply the decision regarding the regulatory decision. Then once you do that, then you might be able to work backwards or reinforce your levels system regarding where it lies and the degree of certainty that that level supplies. and therefore the level of certainty that the decision would incorporate.
I think you're absolutely correct, and I think what you described is a integrated problem formulation activity, which you have not only the risk managers and the risk assessors at the table, but you have a diverse population of stakeholders to flesh out all the possible issues. And then that allows you to walk through what are the key questions that have to be answered. And then what we see as one of the benefits of this approach is once you've clearly articulated that, then if exposure and hazard evaluation is a key feature, then you can go through the matrix. If the key features are policy and politics, then you don't even bother with that. You deal with that first. So I think what you described is really so important, and that's why we start with problem formulation, is if you do that right, then you're focusing on the most important things.
But there is, I'd just like to say there is, of course, one category of regulated substance in food which uses a tiered approach, which is food flavorings, where the TTC, which is an extraordinarily conservative and uncertain approach, but all we know is it's going to err on the side of caution, is widely used to determine whether there's an issue. And only if you exceed the TTC is there any generation of specific chemical specific data. So, but that's because there's so many of them and they're relatively innocuous.
Just because it's a follow-up.
Yeah, I agree with that. I was part of WHO, FAO's, expert committee on food additives, and was there when they instituted that whole approach. And while I agree in general with it, in most cases, there's a good deal of close homology between, the series of compounds in a structural category. Far more homology than there is, say, in the pharmaceutical industry's world. And so what I'm saying is that flavoring materials lend themselves to this kind of analysis more naturally, and utility is there and more easily seen.
Are you through, Hatton? This is what happens when you retire. The freedom is open. I would like to say congratulations, but before it goes to your head, I just want to say that are the people who developed the matrix, which seems to be the keystone to this, are they here in the room? Some of them. I'm right. I'm eating it. My congratulation is that I've always thought that any kind of a leap forward on this is going to be an integration of various things that we didn't see connected before, and that's in fact what Lehman and Fitzhugh Nelson and their folks did back in the 50s. They pulled together stuff that we wouldn't have thought could be pulled together. And it's very simple to us now, but it wasn't then. Let me tell you, from what I'm reading, I wasn't there, by the way. From what I read about all this, people were really confused, and it was only a few people who knew how to do it. But over time, of course, everybody found out. So it seems like you're trying to do that. And I just want to make a suggestion. There seems to be one feature that we're not exploiting, and I don't quite know how to do it, but I'll throw it out. Maybe somebody smarter than me can figure it out. It's what we do, or I've been doing risk assessment for so long, but what we do in doing risk assessment is, the actuarial risk, we're really piss poor at doing it. Okay, but what we're really pretty good at is relative risk. And we sometimes put things in sort of a relative risk framework, but I don't think we're exploiting it the whole way. That's all we really need to do is put things in relative order of toxicity. Okay, somebody's got to draw the line, what's acceptable and what's not, and that's not a scientific decision anyway. So it seems to me that we could, I don't know, make the matrix some sort of a relative risk type framework, and that could get us another boost. I don't know.
Anybody want to respond to that?
We just need to take the shading out of the matrix, and the relative position of the box would give you the relative risk. The relative position of the box would give you the relative risk. It would be two-dimensional because the risk is a product, of course, of exposure and hazard. So the box would move with either one or the other, or both, relative to the other boxes. And that would allow you to compare different compounds or different scenarios, in fact, of the same compound. Would that work? Just take the shading out. And take the units off, yeah. Take the units off. Yeah.
Are you? So are you talking about it as a communication tool of relative risk to other things? So I mean that's no, just relative risk to other.
Yeah.
Okay.
I also want to congratulate the group that is doing this formidable task, but I do have a concern, and that is the quality of the data that you're using. I mean, how are you determining how good is the data? And that should be very transparent. I have to review a bunch of scientific papers to journals now that I'm retired, and there is a lot of junk out there. And so how do you make sure that the data that you're using is good?
Well, I mean, I guess there is always a concern in any risk assessment, and I would contend that the use of that data, of those data, in the RISK21 approach should have the same rigor built into it as it would in any other risk assessment. So that means that you would want a quality criterion for your study. You'd want access to study design and raw data, which would be a problem for many published studies, which is one of the reasons that there's this big debate about using published studies in risk assessment. If it was published, you'd want access to the data. You would want to know that there was a transparent study design. You'd want independent verification of the adequacy of the methodology, and all the things that would go into a normal risk assessment would go into RISK21. Now, one of the big questions, and it's been touched on already today, and it's something we're all faced with, is the reliability of database information. And we've been doing some quality assurance on the databases that underpin the TTC. And it won't come as a surprise that some of the data are not as robust as one would have liked. And how do you curate a 100,000 entry database and make sure that it's adequate? So I think the answer there is there needs to be a set of principles and criteria which the people who construct the database have to adhere to, rather than have to individually go back in and check every single item in that database. I know that we were doing something on the ToxRefDB database, and they were entering new information, and that was a very, very resource-intensive task to individually curate all that information. So I think ideally it should go into data entry, not into cross-checking after it's been entered.
Just
And there
as
should be
a follow-up question to Dr. Sotomayor's question, which might include also the gentleman on risk communication and Dr. Keefe, as we often deal with the petitions from a much more sitting in between two chairs perspective. You mentioned that the tool will be publicly available. How do you make sure that it is not used in various ways depending on where your viewpoint of the world is? If it's a public tool, anybody can feed it the way they kind of like. So are you sure that the right and the left will come out with the same number, feeding it according to their needs?
Well, I'm going to leave Michelle give the official answer. When I said public, I was really just meaning it wouldn't be a private or free tool, but it didn't necessarily mean it'd be open for everybody. It could be, but Michelle can give the official answer.
We could
Screaming
end
at
up
me
with
from the
all
front.
kinds of risk assessments.
Well, Tim likes to use this term that the tool is agnostic to I guess the data, right? The point is that it's transparent, and that if you, it's going to, in the end, garbage in, garbage out, of course, but that it would transparently be able to tell you, this is the data that was used for it, here's the study. One of the things I didn't go over a bit is for any data point you'd mouse over and it would tell you the source and that sort of thing. I think the fact of the matter is there are already a lot of risk assessment types of tools, models that my mom could access if she wanted to. And I think a lot of it is, it's a matter of making it such that I think communicating, and I would be curious to get some insight from you as the risk communicator on this, of what the tool is meant to do, and really sort of transparently identifying those parameters. But again, there's a lot out there already that people can use. I think one of the big things is, it's not to say if you plot your data that you have and your box shows up in the red, it doesn't mean anything without the context, without the problem formulation, without all these other steps. So again, I think the fact that the tool is agnostic, as Tim likes to say all the time, I think that's a kind of a good take home there if you think about it in that way. But I would be curious, I know there's a question at the back of the room, but from the standpoint, I know, Dennis, one of the first things you said was, is this going to make communicating risk even harder? I think maybe naively we were thinking RISK21, maybe this would make it easier. I'm curious from a risk communication standpoint if you have some perspective on that side of things. Dennis or Doug?
Just so that I can, pay for my supper or whatever, so I can say something. When I look at it, my first exposure is to it right now here.
Right.
And so when I look at it, sure. When I look at it, I don't see anything that would make it easier, unless, everybody adopted it and everybody understood how it worked. All the scientists and all the people with competing agendas all adopted it and used it in the same way. Then I think it would be a wonderful thing, because then we'd only be arguing about those things that were on the borders, and we wouldn't worry about what's far in the green or what's far in the red, and it would make things much more productive. But I live in the world of, as the gentleman said early in the morning, I live in the world of theater, and I'm a public communicator, and public and policy, it's very much about theater, and there's competing agendas, and I have to deal with people who have an agenda to push, and I have to use truth, and I always puts us at a disadvantage, So if you can make everybody use this tool, if everybody would use this tool, then that would certainly level the playing field, and it'd be a wonderful thing. But I don't know when that's going to happen, and I'm not ready to throw down my weapons, and so I'm still gonna.
After only 3 hours?
Yeah, I think we still have to, so the bottom line, for me to use this to explain a specific risk scenario to anybody, it doesn't really help me, because it's versatile, and it's not set in stone that this is always going to be green. And so I have to take everything on a case-by-case basis, if that makes sense. And so you know, I feel like I would be starting with develop my communication plan and materials with the specifics of each assessment. And so it might help you explain it to me once you've done it. And that's half the battle, you know, too, is if you can explain it to me, then I can explain it to somebody else. So, but as a public communication tool, I think until we have full acceptance and adoption from everybody, then we're still going to be plowing the same rows.
I mean, I think at the most basic level, I mean, I think there's a couple different levels of communication. I think where we've seen success, and when I've actually given this talk to folks who maybe aren't as familiar with risk assessment, the big take home is that the dose makes the poison exposure component, it's not necessarily communicating this is your risk, this is the number, this is the, you know, the green red, but that it does go a little bit further in trying to communicate the intersection between toxin exposure rather than just being hazard based. But and that's the feedback that I've gotten from a communication side of things so far anyway.
But you know, in any of these situations, I always feel that and again, this is something that I tell people, is no matter what we do, we're going to make somebody angry, right? And so whatever number you have, or whatever, you know, dose and exposure level that you come out with and you feel is safe, there's going to be somebody out there who has something to lose, who might disagree with it, and they're going to, I mean, you're still going to have to contend with them, you know, on this. And you know, if this tool were to erase then, you know, I mean, it'd be amazing. It's really amazing. It's just not going to happen. So yeah, so hopefully that's.
No, that was useful. Thank you.
I guess my point really was that, you know, people, you know, the general public, you know, when they talk to us about communicating to the public, they talk about communicating to the 9th grade level at most, and you know, there's such misinformation out there about the hazards, the risk of chemicals in food right now. I mean, there's a chemophobia going on right now. That a new system in terms of a mechanism to communicate this information to them, I don't know that it's going to help. I wish it would. I think if there was something, there was some way to take this graph, which I think is it's a very nice presentation. It's very, and it, you know, it expresses the continuum of risks that we face. If there was some way of, perhaps for communications purposes, putting it in context of other risks that we deal with daily, like walking across the street in front of this building every day, or taking the metro, or driving on the beltway around here. That, you know, that might be helpful in terms of, for communications purposes. In terms of, you know, what we do every day, maybe not, but something along those lines might be helpful.
Well, I mean, the reason that's maybe a little bit problematic is that one of the things, I mean, there's this idea about if I choose the risk, and whether or not it's chosen for me. And so, I mean, I understand what you're talking about, but, and I've used analogies about, you know, the amount of radiation that you get from this is the same as being on the beach, or taking a flight in the, you know, in an airplane or whatever, but it doesn't always resonate that well. Again, it's about, you know, who's choosing the risk and who's imposing this risk on me, and it's a lot more complex than that. I mean, I think the one thing I think maybe that we should think about is that, you know, it's almost, it sometimes can be sort of like a political campaign, is that you have people who are on your side, or on one side, and people on the other side, and all trying to move people in the middle, you know. And so if the people in the middle, like as FDA, I would say that people trust you, okay? You only hear about the people who don't, but in general, I mean, the people trust you, and they trust the decisions you make. And so what I want to do is I want to keep it that way, and I want to keep, if you tell me that this is safe, then I believe you. I mean, you know, and so I'm going to try and keep the people who are agitating for distrust from moving the people in the middle, you know, and so, I mean, if that's an easier way to look at it, you've got a lot of people in the middle, and so if someone is pursuing an agenda, they want to move as many people in the middle over to that side. Whereas, you know, what you really want is, and you're concerned about safety. And if it's safe, you know, and you feel it's that way, I mean, then I think that the bulk of the American public will trust that. But, you know, there are ways that other people might move them against that. And so that's, you know, part of the explanation is combating those other agendas, I guess, you know, so.
Can I just interject? My favorite risk perception example was I was in a pub in the UK, and you could cut the cigarette smoke in there with a knife, and people were drinking these liter things of beer. But the menu assured me there was no genetically modified ingredients in the food. And so, gosh, I know I felt a lot better.
Yeah, one question. I don't know how simple. So I've looked at the tiered system. I think I've seen it presented a couple times. I think you presented SRA on the tiers of the roadmap, and so I obviously mentioned my concern about the severity, but it almost seems to be a pushing and pulling. I mean, we're scientists and we're trying to extrapolate the most we can out of our information, but then we're coming up against cumulative risk assessment, where if we're trying to do these combinations, I mean, we have so many, and I'm not just talking by oral route, inhalation route, combined routes. We're in tobacco, so you may have a dual user, smoking, smokeless, and multiple components in that. And you're trying to figure out all these impacts. And if you're trying to look at a mode of action or mechanism of action, then you're not in this simple tiered approach anymore. And you're hoping, you know, an assumption of additivity is enough. But it also seems that you need to go to the more complex evaluation to consider all these factors. And so while the tiered approach would be great, you know, considering different routes, different severities, and multiple chemicals, you're trying to balance that. Now, I want to layer that on top with one last, you know, scientifically, this is a simple approach, right? Yeah, that's sarcasm. But on top of that, we have the added that within the statutory authority of our center, we have to communicate what chemicals are in each brand of cigarette so that they're understood and not misleading to the public. So I guess two questions. One, the pushing and pulling of a base tiered system to try and simplify the approach to risk assessment versus the more complex information we need to do these cumulative risk assessments, and then trying to convey this information to the public.
I will just say, having actually been involved in the advice on the toxicity of cigarette smoking, that's not something RISK21 is going to solve.
I think that's interesting, because I think you point out perhaps beyond the ability or beyond where we could go with this right now is complex mixtures, which cigarette smoke is, of a product we know is hazardous. So I think that might be, because if you look where we were starting, it was, and the initial conversations were chemical by chemical, or then, and drugs by drug, yeah, drug by drug. But the other part of that is the communication part, and I think a tool like this, again, we have to kind of go stepwise in that one of the communication is just to risk managers. So from risk assessor to risk manager, and if you're a risk manager, you know that you want to know of all the decisions that you have to make for this particular problem, what are my risks of making the wrong decision? And a tool like this can help visually lay out to a risk manager the different options and frame a conversation. And so these kinds of tools this kind of tool can improve the communication among the knowledgeable. I think over time, that increased ability to communicate easily among knowledgeable folks will eventually trickle into broader abilities of communication. But I don't think a tool like this would be very helpful to communicate to the public. I think it would be contraindicated because you can say, oh, it's in the red, therefore it's bad. No, that's not what then you have a whole nother layer of discussion. So I think these kinds of tools have to be used for what they were intended. Same with all the modeling tools we have available now. You know, your mom can download it, but doesn't mean she should. So
I didn't say she should.
I was volunteered to ask a question. Each agency has its own authorizing legislation and its own way of doing things because it's got its own specific reasons and needs. And so for our Office of Food Additive Safety, particularly in the division that I'm in that deals primarily with food packaging materials, we have a certain way of doing things. And if I can jump forward to the future, there will be beams of light. Packages will, no, that's Disney, never mind. We have a mechanism existing right now where a notifier could put together a risk assessment, and we have two major components to our submissions. There's a brief summary like an abstract, which is called a safety narrative, where they lay out the overall picture of what the safety is for the submission. And then there's a more in-depth, comprehensive toxicological profile where they're supposed to go really into the depth. What are all the studies that support the safety and give you all that background detail that you need that supports that abstract safety narrative. But this is not carved in stone. If a notifier has an alternative approach that they want to submit their safety argument to us, there's no law that says they can't do it that way. And so we would look at, I think I'm safe in saying, we would look at information that was presented in a risk assessment of this fashion. And now that we're educated somewhat about it and with some more experience, that might be beneficial to help us understand and particularly frame the discussion. With my experience at EPA on actually trying to develop a mode of action for a chemical, that is where the rubber meets the road. That's where the toxicologist may have, particularly some of the ones that are more recently trained with the molecular background, but it could be any toxicologist or team approaches using pharmacologists and physiologists and other people that can contribute to a mode of action and lay out the best information they have, connecting the dots, connecting the key events, looking at the adverse effects, and be able to lay out a framework, I think, that could be useful. But that's the biggest part of the work. It's probably got a lot of opportunity for haggling and arguing, like for adverse effects, for example. But I do see that there is an opportunity to lay all that groundwork and to be able to identify where the problems are and then be able to say where you need more data. So I personally would be encouraged, I'm encouraged by this development. I have heard earlier presentations of this and you brought it a long way and hope that it's going to be useful for everyone.
Thank you.
What? Yeah, I know. I'm sorry. It's good. I'm studying under Ron.
Poor Ron.
I think a third dimension to the matrix, if somebody were brave enough to try to put it together, would be to rank order the adverse effects for severity, and even if I can get really speculative here, do some modeling, a sensitivity analysis, if you will, where let that run in a video that is moving from one degree to another degree, from one extreme to the other extreme as to where the risks are.
There was an activity a few years ago, and I think it just foundered, where they tried to get agreement for weighting factors for severity of toxicological effects. And this was not the sort of human health metrics that I was talking about earlier, but actual animal tox. And I think what happened was they just couldn't agree. How would you rank, minor inflammation against, a non-genotoxic carcinogenic effect? It was just, or a reproductive effect, and it just didn't work. But that's what we need eventually. Absolutely, that's where the research grant comes from. I wouldn't disagree.
So as a non-toxicologist, one of the things that strikes me about looking at that matrix is value is for actually being able to identify data gaps and to sort of rank the importance of the data gaps. So if you look at those boxes that you were generating, and as Clark said, almost everything is going to wind up somewhere in the middle where you aren't going to waste any time on it. So by looking at those uncertainties and how big they are and what directions they are, this could be a good tool for justifying which parts of the question need to be addressed most urgently to make those boxes smaller.
And one of the things when we created that web tool, sorry to jump in, but was to make sure that we were able to appropriately visualize on there if you have a point estimate and an uncertainty factor so that you can visually see what part of the box is from actual measured data versus what part is from an applied uncertainty factor, for example, and then helping, again, define those data gaps and determine where to go next.
Okay, thanks for the presentation, everybody. You guys did a good job. Having been doing risk assessment according to the WHO framework, like hazard ID and then hazard characterization and everything, and especially after listening to Ron's introduction, I couldn't help comparing the traditional risk assessment framework to RISK21, and then I realized we have different purpose here. I might understand, right? Because for me, come from food additive safety, we review the safety of individual chemicals, and we automatically goes into tier 3 for toxicity, and sometimes for exposure also. And what is the major advantage of RISK21 towards the traditional regulatory risk assessment?
I'll just take a shot at this. See, my view, when I saw the presentation, I sort of think, oh, these are a bunch of.
Sorry to bug you. We need to.
I sort of think this was designed with TSCA in mind. It's sort of like a TSCA view of toxicology, and it's like I said, in food chemical safety, usually the chemical is a given. So, I mean, I think most of the time it doesn't have that much to do with us. But I mean. We can probably think of some exceptions, but I guess if you're, like I'd say if you're post-market evaluation of food additives, maybe where the exposure. I mean, really, pesticides are really actually an example of food chemicals, and sometimes we do have to do evaluations of pesticides when they're over tolerance, and I guess we might. I guess and actually since they're starting to do more pesticide analyses, you could use something like that to flag which ones they should send to us to evaluate, because they send us a lot of them that should never get to us. So I can think of some exceptions, but as they usually, by the time it gets to us, this is the chemical and we have to evaluate it. So.
Well, I think part of it is like with any new tool, you have to start using it, and if all you have a hammer, all the world's a nail. So there may be situations where this isn't of great value, but you don't know what particular value it might have unless you work through it a few times. And in your particular risk characterization situation where you maybe have all the data that you could possibly need, both exposure and hazard, it's not necessary. But if you start using it, you might find that over time, all that data isn't necessary, that there's key pieces of data that's what you really need. So in the future, what it might help you do is really focus on requesting the helpful data, eliminating the data that is a distraction, because it really doesn't help. So part of this from RISK21 is TOX21 and thinking about the future is how do we get to the answer more quickly? And I think that, to me, changing the conversation about high throughput toxicology is not about robotics and in vitro assays. It's really about getting to the next decision point quickly. So the high throughput aspect is what do we know, what do we need to know, and can I get to that next decision point quickly where I can get the next assay?
I think that was a very good shoe-in to wrap it up, if that's okay. Unless Dr. Sobotka has a question that is very burning. Don't scream. Don't scream.
Don't speak.
Yeah, and I think I have to go back to what Doug was saying, what Jeff was saying. For the table, there seems to be another dimension of severity of the risk that you're talking about, or the severity of the effect of what you're talking about. And you could add a third dimension onto that graph of yours, where you could actually plot that along with the other elements, exposure and dose. And I think that would give you, I don't know, worlds of more information about communicating risk and about deciding as a decision factor. And one of the questions I was asking before, and I'm asking the panel, is there any issues that you see or problems? But there had been mentioned that the ideal is to try to integrate the RISK21 with TOX21 information. But the TOX21, unless I completely misunderstand that, they're focused on internal exposure and not external dosing. And it seems like all of what you're talking about here in the RISK21 is external amounts of external doses. And I'm not questioning why. It's just a question. I don't know whether I'm interpreting everything completely correctly, but is there an issue with trying to integrate TOX1 and RISK21 since their focus is more on internal doses and not external doses?
There isn't, because we visualized the matrix would be plotting like for like, and if you were using internal dose estimated from TOX21, you would also use that for exposure. But in fact, we had a whole sub-team of the RISK21 project working on exactly that question. They've come up with a framework for extrapolating from in vitro to in vivo, for getting the dose metrics right, and how you would integrate that with the exposure estimate. So this is something we didn't present any detail of at all today. It's another dimension which we have addressed, and there are a couple of papers that are about to be submitted on that as well. So it's well taken point, but it's certainly one that we've tried to address.
Okay, going back to the question of the lady over there, how is this different from what we're doing? I think I want to use that question to lead into the workshop that we are planning later this year. And I want you to hold this thought dear in your mind while you're going back to your work and look at risk assessments you've done, how you've done them, and if you would like see them compared to this approach. Sometimes it just helps to, have it done over and find out, oh, I could have done this, coming out with the same conclusion with only, very few data points that you need. So we're proposing this workshop for later in the year, and we'll keep you posted on it. But if you send me in the time course ideas that you might have that you want to be discussed, then I can feed it to Michelle and the group, and they can, you know, have their thoughts on it while we're going. And then we have a very interactive workshop later on to see how it plays out. My suggestion was red 3. I bounced it off of Dennis, but he said he wants to think about it. Because the one danger is we end up with lots of different risk assessments, and I don't know if that's a good thing, you know, as long as we know what we do with them afterwards. So I want to really thank everybody so much today, our panelists for devoting their time and their thoughts and their input for something that they didn't know much about, some of them at least. Our esteemed guests who were so kind to come despite the snow and the flying, and especially Michelle, you know, who made it happen at this time of the year, and everybody who came. So I very much appreciate your interest. Risk might be something that is very disturbing when you read it, but hey, it can actually be fun. So thank you so much for coming, and have a great rest of the day.
And I just want to thank all of you for coming, and for Sabine and Susie for putting this together. We will make the slides available. And if you have any questions in those slides, my email address is there, so feel free to email if you have questions or need more information.